Good day, ladies and gentlemen. We will begin with our third technical session of the event and the first technical session for today. First of all, let me invite our chair and co-chair for this technical session. The session will be chaired by Dr. Raza Shah and co-chaired by Dr. Mohammed Yar. Dr. Raza Shah has already been introduced to you. Let me introduce you Dr. Mohammed Yar. Dr. Mohammed Yar is serving at Interdisciplinary Research Center in Biomedical Materials, Comsec University, Islamabad. He has obtained his PhD degree from Bristol University, UK, and postdoc from Nagoya University, Japan. To date, Dr. Yar has published over 76 journal papers with high impact factor. And Dr. Yar has 14 patents to his name. He's now technical consultant at one of the private companies in Australia, and Higher Education Commission of Pakistan has awarded a grant under the Technology Development Fund. Welcome to you, Dr. Yar and Dr. Raza Shah. With this, I'll hand over the proceedings to our chair and co-chair. Dr. Rashid
Bashir. Good morning, and in some part of the world, it's good afternoon and good evening. It's really a pleasure to start this session with such a wonderful speaker. And we are honored to just welcome Professor Rashid Bashir, who is Dean of Engineering and also Distinguished Chair in the Engineering and Professor of Engineering at the University of Illinois at Urbana Camp. He was Executive Associate Dean and Chief Diversity Officer at the Carle's Illinois College of Medicines. He has published a large number of papers and he has also supervised a large number of graduate students. He is one of the most distinguished researcher in the UK, and recently he has developed a computer, a mobile-based device for the detections of COVID-19 virus. And it's really an honor, Dr. Saab, to have you with us. And as mentioned by Dr. Rashid, that you always accepted our invitation and spare your precious time to share your wisdom with us. So we are very much looking forward for your presentation. It's all to you. Thank you so much.
Thank you very much. Can you hear me okay?
Yes, we can hear you.
Okay, thank you. So yes, it's my pleasure. Thanks for the invitation and then the kind introduction. So I'm at the University of Illinois at Urbana-Champaign in the US. So in the next hour or so, I will talk to you about, so I'm the Dean of the Granger College of Engineering here, but I'm also have a very active research group and still maintain that. So I'll really talk about my research work today and not talk much about our college and my administrative side of the job. So my group's interests are actually broad, and before I talk about that, I want to just kind of really preface by this one slide as kind of a broad interest and a broad opportunity at the engineering, at the intersection of engineering, biology, and medicine. So this is a perspective article that I had the pleasure of writing, Engineering is a New Frontier for Translational Medicine, with three of the pioneers in the field, Xu Chen, Bob Nerem, and Roderick Pettigrew. Robert Nerem actually has passed away since then. He was a great friend and a great mentor at Georgia Tech. Xu Chen is a father of bioengineering at UCSD, and Roderick Pettigrew was the director of the National Institute of Biomedical Imaging and Bioengineering at the NIH. He's now at Texas A&M. So just the idea that there is this tremendous opportunity and a need to really try to bring together engineering and medicine and achieving this vision of higher quality healthcare globally while reducing or containing costs. And we posit that the integration of engineering into medicine and medicine into engineering until these boundaries vanish will play a critical role in achieving these broad and specific goals. So there's a lot that we can talk about what that really means, but I think, really trying to bring in engineering technologies and the engineering mindset. And the engineering, engineering is really a translation of laws of physics and chemistry into practice. The fact that I can forward design, right? Biology is still very discovery based, but we are starting to forward design in biology also. And medicine, which is very observational, the goal is to start to really integrate engineering technologies into medicine, to advance medicine, to lower cost, to increase quality, and to increase access. This triple aim, as it's called, we believe it's, bringing in engineering into medicine is really going to be important to achieve that triple aim. I also would welcome you to look at this report on convergence, the future of health, a report from MIT that is really a very fantastic read on convergence. So with that, let me just kind of jump right into it. And I would certainly welcome the moderator to stop me at any time if you have any questions you want to ask me as I'm going along, and hopefully there'll be some time at the end also. So my group is very interested in using macro, micro, and nano fabrication technologies to then apply these to grand challenge problems in cancer, sepsis, and global health by developing these tools for precision and personalized medicine. That's kind of, I would say, half my group. And the other half of my group is very interested in using these technologies to develop biological systems. If I have time, I can show a few slides on this left side of the things also, but most of the talk today will be on the right side. And let me just start right off the bat with a grand challenge, which is sepsis. And actually, this project, as you'll see, it's really interesting from many perspective, but also that we really brought in data science and machine learning that I'll talk to you about. So sepsis is really a major challenge around the world. It's a lot less talked about. It has a lot less sort of awareness. But in the US, at least, nearly 1 in 3 people that die in hospital actually have sepsis. Okay. Each year, we have nearly about 270,000 people that die of sepsis in the US. The numbers globally are actually very similar, but I just haven't put those numbers here. So sepsis is a life-threatening organ dysfunction due to a dysregulated host response to an infection. It's the leading cause of death in critical care in the US and the world. And just in the US alone, there is over 25 billion sort of cost estimate to the healthcare. It's the most expensive condition and the leading cause of readmission into the hospital also. In the US, there's more cases of sepsis than these 3 cancers combined, and more than stroke and heart disease in terms of the cases per 100,000 in population. Now, it's not to say that other diseases are not important. They're all very important, but it's just that sepsis is something that really, it's not, the awareness is not as high. And of course, nowadays, as you can imagine from COVID, a lot of what happens when someone gets an infection and COVID and the SARS-CoV-2 infection, they actually show the same sort of symptoms and progression of disease as happens in sepsis. So how do you stratify sepsis? How do you decide what to do with sepsis? So in 2016, there was a task force convened by these 2 societies, and they defined sepsis as this sort of what's called now sepsis 3, which I just mentioned also the definition, and it's repeated here. And what is used to evaluate sort of sepsis is this thing called SOFA score, or a SOFA a number. It's sepsis-related organ failure assessment, SOFA. And what happens is that here's a flow chart that's in this seminal paper, which just talks about what it's a recommendation to the physicians that when a patient is suspected with infection, they actually first do what's called a quick SOFA, Q SOFA. And this just looks at 3 variables, and a score is given to each of these variables, and the composite number is what's used. If that number is greater than 2, then you can continue down here. If the number is less than 2, then perhaps the person's condition is not progressing that badly. However, these three numbers, the respiratory rate, mental status, and systolic blood pressure, these can be changing or high. Essentially, the score can be high for a variety of conditions. These are so generic, right? And then when you go down later here, then there's a SOFA score, which has more parameters, including a couple of parameters from the blood also. But still, those are still quite generic and broad, so to speak. So anyway, this is the current procedure. And I'll come back later and I'll show you why this is important from some of the data. So the goal is to be able to see, as patients move through these various stages, that can we sort of develop a predictive capability from knowing the state of the patient early on. Can we predict what will happen later to that patient, depending on their blood biomarkers? That's kind of what we are trying to do. That's our goal. So some years ago, oh, sorry, let me show you this other slide too. So this is what happens today in the clinic, right? Or even before the clinic. You have this onset of infection. You might get an infection. You don't know when you got it. And then it might take a couple of days for someone to feel abnormal. And then, you don't always go to the hospital, right? Sometimes your immune system is strong enough to overcome that and you feel better sooner. If you don't feel well for a while, then you will go to the hospital. You might go to the emergency department. And it might take a couple of hours just to get you checked in and get the vitals and get blood. And then that blood is sent for culture. This is what always happens. However, the blood culture actually takes too long. It takes up to 5 days. The standard is to take up to 5 days to confirm a negative. So the physicians can't wait 5 days for a blood culture. So what they do is they prescribe these broad spectrum antibiotics. And actually there's all sorts of issues with that. We are over-prescribing, as you know, and giving people too much antibiotics in general across the world, wherever it's available, of course. So after that, the blood culture results might come out. However, whenever they come out positive before 5 days, but they have to wait till 5 days to confirm a negative. Then it might take another hour to do an identification via PCR, another day to do some antibiotic susceptibility testing, and could take many days for the patient to get better or not. So this is kind of the broad workflow. This blood culture takes too long, and I'll talk about that a little bit later. And 50% of more come out negative, actually, because it has to do with sampling the blood. You take 5 mLs of blood, and you might not get the pathogen in that 5 ml of blood from the draw. How do you prioritize and stratify the antibiotic treatment? How do you know who is important to prioritize? Because the busy hospitals get a lot of patients at the ED, and you just can't treat everybody the same as they come into the emergency department. And how do you quantify this profile, the profile of the patient immune system over time? Because that's really what's really important. So we started a study now a couple of years ago, but it's still sort of continuing and growing by sort of doing what I would call precision medicine approach for sepsis. We pulled a big team together with our partner hospital in town, Carl Hospital. So all of their sort of the key physicians and the ED director and all, our group, which builds a sensor for a living, sensors. We also brought in some collaborators in machine learning and predictive analytics, and also colleagues from biology, sort of from the disease biology aspect, and pulled a big team together. There's multiple papers that came out of that work, but I'll show the summary. The bottom line is that we actually collected blood sample for every patient for which a blood culture was ordered. Okay, so every patient blood culture was ordered. We also get a tube of their blood. And we would measure all of these parameters right here. We would measure these proteins, the pro-inflammatory proteins and the anti-inflammatory proteins. This top one, neutrophil CD64 and monocyte, are actually cell surface markers. And then we also obtain the clinical information for the patient for the entire stay. So we get the blood sample at T0 in the beginning, the first blood draw that was taken. We get a sample of that. And then we get the patient stay through the entire, stay of the hospital, essentially. So I'm just turning off my phone here. Okay, so then the idea is to take all this data and we actually put it into, we developed a machine learning algorithm. Now, however, to do that, there has to be an education process because there's no controls right now. There's no gold standard. So how do we know what state the patient really is in. So working with Dr. Karen White, who is the director of our ICU, and Ruoxing Zhu, who's a biostatistician, machine learning expert. This slide is a little busy. There's a lot of text, but I'll just talk about the summary. We basically looked at 5 different machine learning algorithms, looked at with and without various feature selection, and then we had 10 physicians and residents that actually looked at all the charts of the patients and put them in these categories, essentially. Patient that might never be septic, and then essentially these 11 categories, okay? So that became a way to do the adjudication for what might be happening to the patient, at least trying to develop a reference check, essentially, for the data that we were collecting biologically, right? And then this is kind of the outcome of it. Long story short, we get these normalized feature coefficient on the y-axis, and here the x-axis shows all of the parameters that we get. These parameters on the left all the way to CD64 are parameters that we measure, whereas these other ones are essentially what typically you get from the electronic medical record, okay? So what this shows is that IL-6, which is a protein, PCT, MMP9, for example, these 6 you can see, play an important role, right? So does it shows pulse and CO2, but however, those are of course extremely non-specific, right? But these features right here, correlate highly to the outcome of the patient, essentially. So this is what this shows is that sepsis is you don't have you won't have one marker for sepsis like you do, for example, for a heart attack, right? But in this case, there's a panel of markers, and that we should be considering measuring, multiple markers, multiple proteins for sepsis from blood. Now, what's really interesting, and this kind of is the bottom line here, is that when you do this AUC curve, looking at the predictive power, true positive on the y-axis, false positive on the x-axis, we want the area under this curve to be 1. And it's really interesting. Look at these two, the orange and the purple line. This is the current standard of what happens in the clinic with that SOFA and QSOFA score. So when you look at that, your area under the curve are 0.4 and 0.5, which is like a coin toss. So this is what the physicians are using today. Essentially, it's just a coin toss. Like you can see there's no accuracy to this. However, when you start adding the various parameters, your predictive power goes up. And this last one here, when you add these 5 or 6 parameters, you can get up to 0.81, which is pretty good. And actually our most recent data in this most recent paper shows this to go up to 0.9. So this is really exciting. What this says is that if you can measure all those parameters and use our machine learning algorithm, we can actually predict the outcome of the patient, reasonably well, or at least much better than an AUC of 0.5, which is essentially, like I said, a coin toss. And what's also important here, which shows is that this is x-axis is hours of the EMR data used. Y-axis is the area under the curve, which you want it to be as close to 1 as possible. And what this simply shows is that if you take the EMR data only, you can also get better predictive power, but it will take you up to 16 hours of EMR data collection to get the same predictive power as the proteins from a drop of blood at time t0. So essentially at time t0, you can get the same predictive power as essentially, you'll have to collect 16 hours of EMR data. So this shows does a comparison of, EMR alone can also be very useful. And there's many companies that are actually working on that, trying to just use EMR data only, versus we are trying to use the EMR data plus the information from the proteins from the blood. So this approach actually has been, for full disclosure, we have started a company a few years ago called Prenosis. And Bobby Reddy, who was was a postdoc, PhD student and postdoc in my group, he's a CEO. And the approach now is to actually build a big database. So this Gnosis data set, build a big database of thousands of patients by partnering with multiple hospitals. And the idea would be then you have the big database, and when the next patient comes, you use a point of care device to measure the panel of proteins. And then by measuring that data and comparing it to the database via an advanced machine learning algorithm and a tool, this tool will then guide the physician, inform the physician of what the probability of the outcome would be based on that big data set and based on that patient's blood marker at that time. So this, we believe, would be very useful to try to prioritize which patient should be treated first or earlier. The company is moving forward, and I think we hope that this would hopefully be able to have an impact on patient care. Now, I wanted to give that preface because that was something new for us in the last 5 years or so, where we also got expanded into data science and machine learning. But now moving forward, working through the other things we do, the idea, was that the machine learning algorithm tells us which parameters are important to measure. So then you would actually develop a point of care device to measure those parameters. That was kind of the thinking, right? Let me talk about now point of care in general and a bunch of other technologies that we're working on. The idea of a microfluidic lab on a chip that can, measure pathogens, potentially measure proteins in cells, be able to program or design this chip differently by putting in different modules for sample processing, for molecular recognition, for perhaps amplification on chip and sensing on chip, right? So that's kind of a broad vision. But there's two things that I'll talk about how we are trying to use microfluidics, or at least some approach that are inspired by micro nanotechnologies. But the two things you really need to solve at from a broad perspective is this bacterial culture, or culture itself. I said bacterial culture here, but really culture. And then the other thing that is done in the lab a lot is hematology analyzer, which is a measurement of blood cell counts and the types of blood cells. And also a flow cytometer, right? Flow cytometer can actually measure cell surface receptors and circulating proteins and even small molecules. So we like to be able to see if how much of these functions can you put on a point of care device in a microfluidic device. Okay? So let me give you some examples. So let me talk about this part first, where how can we measure different blood cell types? So for example, going back in the example I just showed, there was a CD64, for example, cell type. Well, that's a neutrophil, that's a blood cell with a specific surface receptor that you can measure in a flow cytometer, but can you put that function on a chip to make a portable device? So here is a platform that we have been working on for actually quite some time and published many papers on that. We take whole blood, we can lyse the red blood cells, then we can actually introduce a quenching buffer to stop the lysing, and then we can have the white blood cells go through a channel where they can be electrically counted, basically a Coulter counter on a chip, which is not new in itself. That's been published before by us and others. But we then did something new, which is to add a region here where we attach antibodies for specific cell capture. And those cells can get captured, and then we count again electrically what's going out, what is not getting captured. And the difference of these two counters can tell you what's been captured. So you can actually kind of combine the functions of a hematology analyzer and a flow cytometer here on a chip by actually trying to measure a limited set of parameters. So for example, this is a chip from many years ago, but essentially we're still continuing to sort of improve and add functions on this, where you can see here, this is blood flow, essentially through a microfluidic channel, and the red blood cells are getting lysed. That's why the solution starts to get transparent after some time, because the red blood cells are getting lysed. And you recover the white blood cells. If I was to stop the video here somewhere, you might see a few little spots here. Those that right there, there's 3 right here. Those are the white blood cells, essentially, because white blood cells are a lot less in number. So then the solution becomes quite dilute, essentially, when you lyse all the red blood cells. And this is what the chip looks like right here, holding fingers with the 2 counters, the lysing module, quenching module, capture. So with this approach, we have published on measurement of white blood cells, lymphocytes, monocytes, and granulocytes. We cannot distinguish between these two for now because their electrical properties are very similar, but essentially you can get a combined count. And then we have also shown and published on CD4 positive, CD8 positive, and CD64 positive cells, white blood cells. And also very exciting, recently we've also shown that using the exact same platform, we can also measure protein biomarkers. So I'll try to skip through this actually. This essentially is an electrical Coulter counting data from the two counters. You get this histograms, essentially these a bunch of pulses, and you can subtract these populations from each other to get the cell counts. The cells are captured in a region with a high surface area to volume ratio. Essentially, these are these pillars that we fabricate in PDMS. This is about 60 microns high and about 40 micron diameter of the post. We attach antibodies to these surfaces. We functionalize the antibodies to these surfaces, and we can get cell capture as cells flow through this. And we've done a lot of modeling of trying to model where the shear stresses are highest versus the lowest. And here is a picture, two pictures shown from the top. This is a top view of CD4 positive and CD8 positive cells, but you can see these bright spots are the cells that are captured on these posts. And we can get very high efficiencies of capture. We also demonstrated this, actually this was my PhD student, Omar Hassan. You might have heard of him also. He's now, he's a professor at Rutgers, an assistant professor at Rutgers. It was his work, so he was the lead PhD student. This is showing the CD64 capture from patient samples. This plot on the left shows the control normalized expression. And when I say control, this really means it's from the flow cytometer, which is the current big machine that the clinical labs use. And the y-axis here is from our chip, from patient sample, from 60 blood samples from patients. And you can see we get a pretty good correlation here, as taking into account that these devices were fabricated by graduate students and undergraduate students in our lab, in our clean room, and then they were tested in our lab. And you can also use this type of devices to monitor the count over time, which just as an example show here, we actually then tracked a couple of, multiple patients here, here as shown, and you can kind of, monitor their progression or changes over time also, which would make that AI machine learning algorithm that I mentioned earlier even more accurate over time if we could have blood measurement data over time. So, we can do we can capture cells and count cells. Now using the same exact platform, we can also measure proteins. And the way we do that is to actually use beads, because now, we have a particle counter, basically what I showed, and we have a way to capture the particles. So what we do is we take beads, functionalize them with the target protein that you're trying to capture, mix them in the sample, and then the beads will, capture the protein, and then we attach another marker essentially on the post, so that only when the target is present, these target molecules are captured and are present and captured, only then the bead would get captured in the post. And again, we do the electrical measurement to count the captured beads. So using that approach, which we've also published, we can measure PCT. So this is PCT with increasing concentration. We can see, and then we can also measure IL-6, which were two of the proteins that were in that panel that I showed earlier. And again, we can differentiate between high and low PCT in the background of the other protein. And then most recently, we've collaborated with Professor Hee-Sun Han here in the chemistry department. She's an expert in microfluidic based hydrogel bead fabrication. So what I really wanted, we challenged her that, hey, can you make beads for us that have a different electrical signature without changing the size? So if we can change the size, we can obviously get a different impedance in this aperture as the beads pass by. But can I make the same size beads but get a different electrical signature? So she actually did that by working with our students, and her student, Michael Cole, and our students worked together to introduce magnetic particles within this polymeric beads. And by changing the size slightly and by changing the composition of the gels, we can actually get a different electrical signature. So we published that also last year, where different beads, as MHB1, 2, and 3 are 3 different beads. And here you can see that we can get a distinct signature from 3 different beads that are about the same, close to the same size. So very different pulse height in this case. Electrical signal is very different. So now you can measure 3 different proteins this way, essentially, from the same sample. So that's what we are now working on improving. But our paper from last year showed that we can indeed distinguish between IL-6 and PCT, for example, shown here. So this basically shows that using that same platform, we can also now measure, proteins. Okay, I'm gonna keep moving forward. I guess, should I take any questions now or keep moving forward? Okay, I'm gonna zoom to just keep moving forward to save time. I can come back to the questions. So the other thing I wanna mention is this bacterial culture. So this is, or culture broadly speaking, this is a major challenge. I think if anyone can solve this problem, essentially, which is to eliminate the time for culture and be able to detect multiple pathogens at the same time, that would be phenomenal. But let me tell you, kind of just give you a quick background again of the current practice in the clinical labs when it comes to that. It's I think in many of these projects, and it's really important to understand the real clinical practice and the real need, because many times us engineers want to invent things for the sake of inventing, and there's many things that we invent that are not useful. But I think it's really, really important to really understand the clinical problem and then find the right solution for that. So for bacteria, what happens is that if you go to the hospital and what they would do is they might do a venous draw from your arm, 5 ml of venous draw of blood, and maybe sometimes they take multiple tubes of 5 ml. And then with that, what happens is that there's a culture that's done for up to 5 days. And I won't go through the details of culture, but these are culture bottles with oxygen, and they could be gram negative or gram positive, favorable conditions. But you do a culture. Now, the beauty of culture, bacterial culture, is that the bacteria grows. And the bacteria that is culturable, by the way, there are also non-culturable bacteria. But the bacteria that are culturable, they actually divide every 20 to 40 minutes. For example, the most typical ones, E. coli takes 20 minutes. Listeria monocytogenes takes 40 minutes to double. But the point is that if you do the simple math of doubling, in a couple of days, you can get millions to billions of bacteria. And that then allows you to have a very high a high number in that 5 ml. And then I can take a small volume from that 5 ml. And every small volume I take, which is maybe about 20 to 30 microliters even, that then I would run through a PCR machine. In that 20 to 30 microliter, because the concentration has become so high, I'm bound to catch a few. And that's why then I can run then a small volume of a few tens of microliter through, here's 2 standard machines. One is from Cepheid, one is from this BioFire film array. Then I can take that small volume and do a sample prep still and run PCR, right? And that could take a couple of hours. Now, because the volume is so large, I have 5 mLs available, I can run 20 different reactions at the same time by taking 20 volumes, 20, as I said, 20 microliter volumes, right? So this is not really multiplex. It's called a multiplex PCR, but technically it's not multiplexed. You're just running multiple reactions in parallel, right? And so this way you can detect up to 20 pathogens. This BioFire film array has a kit for 20 different pathogens, but there are 20 reactions run in parallel. I hope that makes sense. Viruses, let's talk about COVID or HIV or any other viruses. Viruses there's no way to grow the virus in the clinic easily. Viruses need a host to grow. So typically you need a cell line to grow the virus. And you don't do that in the lab. It's too time consuming in a clinical lab. So what you do is you do a 5 ml of venous draw, and all you could do is when you take 5 ml, if your concentration is 5,000 particles per 5 ml or higher, now, even if I take 20 to 30 microliters, I'm going to get a couple of particles in it. And then I can run the PCR in those, okay? So this is why there is sort of a much higher detection limit for viruses than it is for bacteria, because bacteria go through a culture step. Viruses do not go through a culture step in the clinic, okay? The third thing that's done in the lab, but not as much less frequently, is fungi, yeast, and molds. And they're also there. You can actually do a culture, but believe it or not, they take even longer to grow. It takes 2 to 4 weeks. So essentially, again, you can't wait for that. You could, and if you do, then you have a very high sensitivity. But if you can't wait for it, then your sensitivity would be similar to what it is for viruses. So I hope this makes sense. Long story short, there is 2 companies actually only in the last few years that have gotten an FDA approval. One from T2 Biosystems, one is called Qvella. What they do is they take 5 ml of venous draw, and then from the 5 ml, they have a procedure to directly capture the bacteria using beads, using functionalized beads. And then once they're captured with the beads, they can isolate those bacteria and then run the sample prep and PCR. And they claim to have, be able to do one pathogen in, let's say, up to 6 hours, okay? So these companies have very high valuation. They got FDA approval and they've raised a lot of money to build these products, but they can only do one pathogen. However, they can do it in like up to 6 hours max is what they're claiming, and we have verified it. So if someone in the audience can come up with a way to use a few hours, but be able to detect many pathogen, let's say 15 to 20, then I think that's a real winner. Okay? I will just show you what we are doing here. We have a paper in review in PNAS, but I'll just show this. This is not published yet. We were inspired by the dried blood spot technology. This is commercially used today, where you can put a drop of blood and actually on this filter paper, and the viruses or bacteria that are in it are actually captured in the filter paper as the blood dries. And you can even ship this paper in the mail, essentially, in a package, essentially. So this is how, a lot of times samples are transferred actually from small volume of blood. It's called the DBS technology. So we were reading about that and we said, hey, maybe we can scale this up to larger volume. And is it possible? So that's what we came up with. I'll just show you our process, which works actually within 2 hours. In this case, we were taking about a 1 ml of blood, which is the volume used for pediatric cases. And then we have a process to concentrate that. Then what we do is we basically heat dry their entire volume, and we add our reagents for amplification of the DNA for that bacteria that you are looking for. We go through this step of a thermal lysis. And what this does, it creates a biphasic material. Actually, it creates a solid matrix. So dried blood looks like this in an SEM. This is a dried blood layer. And it actually becomes kind of a micro nanoporous when you dry the blood. So we sort of optimize that, and this material has a porosity. So the bacteria would be stuck somewhere inside this dried blood. And when you add the reagents, it turns out that this dried region still stays solid, but it's hydrated. So it's a porous material that the reagents can go inside. And then there's a network that's built in just simply because the way you dry. And you can actually then get amplification of the target bacteria. So we show here, this is E. coli in 800 microliters of blood. Within 2 hours, we can detect, and this right here is a threshold time for amplification of the DNA target. And here you can see, even at 1 CFU per 800, one bacteria that was spiked in that volume, we can see that we can get 5 out of 8 of the experiments were positive. So you can easily detect in a few, let's say, tens of pathogens, even down to 1 in 800 microliters. So this is quite exciting, and we're gonna see how this moves forward, but we're continuing to optimize this approach. Okay, let me now get to SARS-CoV-2 and talk about that. So SARS-CoV-2, sorry, this is a very busy figure, but we're just submitting a review paper on the detection technologies for SARS-CoV-2. So there's different sample sites, of course. You have the NP swabs, you have the nasal swab, nasopharyngeal swabs, and also saliva potentially. There's 3 tests that we talk about when we talk about SARS-CoV-2. There is what's called the molecular test, which looks for the viral RNA. That is the gold standard, okay, where the target is amplified. This test is very sensitive. It's the gold standard. It costs, the raw cost of doing this test, by the way, it's about $15 to $20. The sort of the raw, the base cost. Then there's, of course, depending on where it's being done, there are premiums added or the hospital cost and all that. And it could take 30 to 90 minutes based on what technique you are using. But that doesn't take into account the time it takes to get the sample to the lab and the time it takes to report out the data, right? That could take many days in some places, depending on how busy the lab is, but certainly it could easily take 5 to 6 hours to get the results back. The other test we talk about is what's called an antigen test, right? So the antigen test is still looking for a virus, for the virus, but in this case, it's looking for the proteins from the virus, right? So it's most always this S protein, the spike protein, or maybe it's the N protein. But in this case, you're looking for a fragment of the virus, right? And this inherently antigen test is less sensitive because the target is not amplified, okay? You're not, unlike the RNA going through PCR or LAMP, you can actually get millions to billions of copies. In this case, you're not amplifying the target, so fundamentally it'll be less sensitive. However, this is more available at the point of care using these lateral flow immunoassay strips. And the cost is maybe less than $5, and it's certainly fast. It can take 5 to 10 minutes. So right there, you can see also that for SARS-CoV-2 or for any amplification-based test, what you're looking for, that can you come up with a 5-minute PCR or LAMP test that is also low cost. Even if it's not low cost, if you could match this time for the antigen test, then nobody will use an antigen test after that. Antigen tests are used because they're fast and because they're cheap, but because they're fast. And everyone knows they're not as sensitive. The gold standard is still PCR. And of course, we also talk about detection of antibodies. That is measured from blood, venous blood, or capillary blood, like finger prick, or actually a draw. And in this case, you have enough protein, enough antibodies in the blood that sensitivity is not an issue. It's the same technology used here for this one. And this, again, is also available at point of care. And, this is something, of course, as you know, to check immunity, whether you have immunity, whether you got vaccinated or whether you had COVID before. So we have been working on viral load on a chip, viral measurements for actually many years. And we have published papers on HIV, HCV, Zika, dengue, and chikungunya viruses. Some of it work was from my lab alone, and some we've been collaborating with Professor Brian Cunningham here in the EC department for many years, and also with Professor Bill King here, who's in mechanical engineering, who is an expert on additive manufacturing. So the approach is kind of what we have published in the past is to make a cartridge which can take a drop of blood. This was our prior work was from blood. Take a drop of blood, lyse the red blood cells, and essentially collect the solution and run amplification reaction using LAMP, which is an isothermal process, and then have the cartridge be inserted in a small reader. And then you have a heater in here that then allows us to heat the sample. And then we use a cell phone camera to measure the fluorescence to see if the reaction is happening or not. So using that, we can kind of get data like that, which we have shown the positive control against negative controls for these different viruses. So we were able to pretty quickly adapt this technology to SARS-CoV-2. The genome was published on January 10th and 11th. Essentially, that week, January 10th, 11th is when the full genome sequence was put out by actually the Chinese government. One of the Chinese research labs had published that first. And many groups, including ours, we immediately, as soon as the sequence came out, we developed our primers. And then in the next, couple of months or so, we were able to sort of develop this test. And this is what actually FDA did too for PCR. What I'm showing you here is what we did for LAMP. So we would take, in this case, the nasopharyngeal swab is put in a viral transport media. And there's a protocol for this. And then what we do is we actually take half of that solution and run it through the lab, the clinical lab in the hospital, sorry, down here, which is the RT-PCR pathway. And then the other half, we run it through our RT-LAMP process using our primers that we developed. But over time, there's really multiple groups came up with similar primers. We were able to do it in parallel. And here you see data here, which shows positive, negative in the clinical samples. The y-axis is fluorescence intensity, and the different colors here are time. So this is 10 minutes, 20, 30, and 40. And what you see here is that, the negatives, there is a background fluorescence, but then as you run the reaction, within 20 to 30 minutes, you can very clearly show the difference between the positive and the negative. And that is obtained from the cell phone camera that is viewing essentially this cartridge and these reactions right here. So this was Professor King's student helped us. We designed this. The chip is similar to the one we did before here. This was a silicon-based chip that our students fabricated. And this time we were able to produce this additively manufactured 3D printed cartridges, which has these two ports. There's a mixing region, and then the solution goes into this flower-shaped region where the solution is collected and it's amplified, and we can watch the fluorescence using a cell phone camera. And this paper was published then later this summer. We put it on Med Archives early in the summer, and it was, or like midsummer, and it was published in PNAS later, sometime in fall. So this was actually pretty exciting, and I think we're actually continuing to move this forward to see if we can further improve this. We also then collaborated with two of our hospital partners, and this paper is an archive and hopefully should be out for review soon, where we further optimized the RT-LAMP process to make it even more sensitive by adding a two-step essentially process by splitting up the cDNA synthesis step. So using that, we improved the sensitivity of LAMP from VTM and from saliva. We also actually then measured this from saliva, and it works quite well. The standard RT-LAMP, the one-step RT-LAMP, which is still reasonable, allows us to go to, let's say, 4 times 10 to the 10, 40 particles, viral particle per reaction volume, or sorry, per microliter, 40 particles per microliter, which is very sufficient for most clinical applications. But then with this improved process, we can actually go down to just a few molecules. And again, this sensitivity then is very, very similar to the PCR sensitivity. And here is some data we did with patient samples, looking at positive the sensitivity, specificity, and AUC and all. So in general, I mean, this is all done in the lab, but the data looks very promising. So we are trying to figure out how we move this forward towards commercialization and FDA approval. Okay, let's see, how much time do I have? Do I go a little bit further, or shall I stop to take questions? Because I have a few more slides, but I can stop now if you think the time is going to be up.
Dr. Rashid, you are having 5 more minutes.
Okay, 5 more minutes. All right, so I'll try to just show you this one new sensor type that also we're pretty excited. And I'll try to go to this a little quickly if you don't mind. But this is basically the idea of using a field effect sensor to measure a biomolecule by measuring the presence or absence of charge. So, DNA, proteins, they have a net charge on them, or they have an intrinsic dipole that could be measured, or a charge perturbation can be measured when you bring the molecule close to a silicon transistor. So you use a field effect sensor, and this is how there are commercially available pH sensors. So point being that you can measure the binding of binding of molecules an antigen antibody measurement or a DNA binding event. But one of the problems with FET sensors is that the counter ions in solution can actually shield the charge. So as long as your molecule is within the Debye length, or enough of it is within the Debye length, then you can actually measure the charge. If the molecule is past the Debye length, longer than the Debye length, then it will not be able to be measured. The Debye length of a sort of standard physiological solution is actually 10 angstroms. Okay, 0.1 molar salt is 10 angstroms. So anything within 10 angstroms, you can measure. Anything more than 10 angstroms, you're not going to be able to pick it up because it's neutralized. So long story short, there was a paper actually published by Professor Chui at UCLA that was just a computational paper which projected that if you could make a concave versus a convex sensor, then you might have a difference in the Debye volume and the Debye length. So the screening would be different if you have a curved surface. So using that idea, we actually developed a crumpled graphene sensor. And this paper also actually was published before in terms of attaching graphene to a polymer layer and then shrinking it so that the material shrinks and the film is crumpled. And this is through a NSF MRSEC center that we have here. And Professor Oluru, who is our close collaborator of ours, we asked, can you simulate this and see what's happening? So he actually did molecular dynamic simulation of individual molecules on graphene with ions and showed that indeed the Debye layer is different on a concave surface versus a convex surface. Okay, and this again was shown by the other paper also. So we then made devices, and this paper was published past year in Nature Com also. Long story short, when you have these curved surfaces, these crumpled surfaces, you get both concave and convex regions. And it turns out that then the Debye length, the Debye layer is extended. So you get regions where the molecule is not shielded and hence it can be detected at lower numbers. Also, it turns out that when you bend graphene, theory has already shown that you could potentially open up a band gap in graphene, which then makes it semiconducting, which can also essentially provide a large change in current. So using all of that, we were able to actually show that we can get some really very high sensitivities down to attomolar, hundreds of attomolar, maybe even tens of attomolar range using this crumpled graphene sensor. So this is a pretty exciting new device, and we have now most recently unpublished, this data is not published yet, but we can also detect actually COVID S protein and N protein, the spike protein, using this technology at very low concentrations using, so this will be the antigen test essentially, that could be a very sensitive test. Okay, with that, I will actually stop and I will go to the end of my presentation. Sorry, I'm gonna skip through this. Just thank my group, a wonderful set of postdocs and students over the years that has certainly helped do everything we do. Phenomenal students and postdocs over the years, and then many collaborators. We have a very highly interdisciplinary group. The top line here is faculty at UIUC. The first top two lines that over the past 12 years I've worked with in various papers. This is our collaborators from Mayo Clinic that we have published before. These are still colleagues and friends from Purdue. And then also we had an NSF center with MIT, Georgia Tech, and Illinois, which also had, the work I didn't show. So with that, I'll stop. Thank you very much, and I can take questions.
Thank you very much, Dr. Rashid Bashir, for such a wonderful lecture. So house is open for questions.
There's a couple of questions in the chat. Shall I just answer them, or do you want me to read them and answer? Or
we can just get questions from the audience and then from the chat. Huh?
Sure.
Thank you very much, Dr. Rashid Bashir, for a very informative talk. So I actually am very interested to know your views about like integrating engineering along with the basic sciences and chemical sciences within the medical curriculum. So how do you think like teaching biology and medicine through the lens of engineering can be helpful?
Well, that's a great question. And actually, I don't know if you all know about a medical school that we have started here at the University of Illinois. It's the world's first medical school, which has engineering as a pillar. And I was actually fortunate to have helped start that when I was the head of bioengineering. So actually, I would encourage you to go look at Carl, Illinois College of Medicine. And it's the world's first college of medicine where what we have is for every course, we have 3 course directors, a basic scientist, a clinician, and an engineer. And our hypothesis was that just like you can't be a physicist without knowing math, we believe that in the future, you can't be a physician innovator, physician leader, unless you know technology, engineering, and data science. So we actually developed new material that integrates those 3. And we have it in a new medical school that's very new. It's only been 3 years. We only have our 3rd class that's going through it now. Like the first class is going through the 3rd year now. And we just achieved provisional accreditation for this school.
All right, thank you so much. I have another question. Okay, so I went through your work and I just have a question. So I would like to ask you. I want to ask you, do you think that the microchips can be sent into the body and they can be powered by the body's natural temperature? Because as much as I know, when the microchips they are sent into the body, they can be very useful and gather a lot of vital information. So do you think the power can come from the external and the internal source? So my question is that if you want to have the chip for the long time in the body, can we choose the temperature of the body as the power source?
Yeah, yeah, that's a
Since
very good question. Yeah, sorry, go
this
ahead.
energy transformation, like from heat to voltage. So
Sure,
is the
sure. Yeah.
transformation of the temperature possible?
Yes, it is, and it's been published already. So that's actually, there is some initial publications on that, and there's a whole field of energy harvesting. I don't work in that, but there's many papers and people around the world and in the US that have worked on that, whether it's getting energy from vibration or from other ways of getting energy. So using temperature 37 alone, if you remember, I mean, if you look at the basic, you need to create a differential, a constant temperature differential to get energy at a constant temperature. So from a 37 to room temperature, that change potentially, if you can somehow do it from inside the body, the temperature to the outside. Yes, that's a very exciting area, and I would strongly encourage you all to look at that. And it is very possible. Scientifically, it is possible. There's also just the various chemicals that are present and the chemistry of blood. There's some very interesting possibilities to actually generate energy from within the body. So I don't work on that, but I think that's a very exciting area.
So I have a last question. So I also want to ask you if do you think that 3D printing that can be used to make the PPE, personal protective equipment, like the clothing, gowns, and face masks? What do you think about that? Is or there are, because there are some technical challenges, I think.
Yeah, actually, that is also possible, and it has been done. Actually, when the COVID had hit, many places around the world have demonstrated that, including our own university. I can send you a link if you send me an email, and I can actually point you to that. So there were faculty. I wasn't involved in that myself, but we had charged a big group of faculty from civil engineering and the College of Medicine, the Illinois College of Medicine, that worked together to do 3D printing of masks and other things to generate the initial prototypes. And it has certainly been demonstrated that that's possible.
All right. Thank you so much, Dr. Rashid, and probably we can talk in detail some other time. Thank you.
Okay. I have a question. First of all, thank you very much, Professor Rashid, for your nice and innovative talk. My question is concerning regarding the genetic variations in some individuals. So how your data can be validated regarding that genetic variation? And second part of the question is that commercially we need to somehow like produce all these like magnetic beads in a large scale. So would you somehow like suggest to go for like 3D printing or some other technology? Thank you very much.
Sure. So to answer your first question, I mean, genetic variations, again, yeah, there are many technologies that are used. for that now. The work we showed, if you know what sequence you're looking for or what region you're targeting, and we can develop the primers against it, we can do that. So essentially, as long as you have the sequence, the sequence that you of your target, and the region that you want to target, we can design probes to do that, right? So that's actually pretty standard. It's very doable. So that's how one would look at genetic variations of the species or whatever else. So for example, in the SARS-CoV-2, the D117, the way it's detected today in the clinical lab is to look at the the S gene, the gene for the S gene versus the N gene essentially. And there are differences in that. And if one is picked up, it's one strain. If both are picked up, it's the other strain. So that is very possible if you know, as long as you know the sequence. To answer your second question, there are actually many, I think, commercial sources now for microparticles and beads, and there are even companies trying to make nanoparticles and have made nanoparticles. Most of the work we showed with particles, we use either purchased beads or we created these beads in polymer hydrogel beads, working with Professor Sun. Where she used a microfluidic device essentially to synthesize to create the beads using a droplet technology. So there's a range of technologies there, and I think depending on what kind of beads you're trying to make and for what application, I think the technologies are out there now, and there's certainly room to improve. One of the main things with nanoparticles or microparticles might just be the scale up of the technology. How do you make lots and lots of them in a reproducible manner where the size and the composition is very uniform?
Okay, Dr. Rashid, there is also in the chat room. So
Professor Reza, may I ask a question?
I would just pick randomly one. There is one question from Aisha Amna. In case of dried blood sample, how can we target the microbe, as there are many microbes which can produce similar type of infections? Is the primer are specific or it is universal?
No, so whether you use dried blood spot technology or not, you can detect different microbes. You can, because of the different primers you use. So yes, the primers are specific for specific bacteria. So for example, you can detect E. coli versus Listeria versus MRSA. So yes, and then whether you use a dried blood spot technology or not, that doesn't change the fact that you can target specific microbes for detection.
I was told that Dr. Animbar Sab is online and he would like to ask questions. So. Dr. Sab, can you turn on your mic? Yes, control.
Hello, Dr. Rashid, welcome to Pakistan. It's a long time I met you in Illinois, you remember?
Thank you. Yes, I do. Thank you. Good to hear your voice.
Okay. We are these days interested in the TN-alpha inflammation reduction using nanoparticles in cardiac patients.
Okay.
How sensitive is your microfluidic chip for these studies of TN-alpha?
So we have not measured TNF-alpha, but I think we can using this approach, at least the microfluidic bead-based approach that we have, or using the graphene sensors. As I showed, we have measured IL-6, PCT, and some of the COVID proteins, but we actually have not measured TNF-alpha yet. I don't see any fundamental issues as long as you have an antibody present. Our technologies can detect, down to the bead-based technology. Electrical counting could detect down to, picomolar range, and then the graphene sensor was very sensitive. It's very sensitive. even in serum, we were able to detect molecules down to, as I mentioned, sub-femto in the attomolar range.
But in one of your earlier slides, I did CET and alpha measurement along with several other proteins.
That's a good observation. That was done using today's current technology in the lab, using a bead-based Luminix assay that's commercially available. So first, when I showed that the big study that we did, there we use standard instruments in the clinic to collect the data. Yeah, and then the on-chip work that we did was what I showed was PCT and IL-6 only at this point.
Okay, my last very quick comment is regarding you are talking about millions of cells analysis and different types. The high energy physicists for years have been searching different programs or different particles, millions, 1 in a million and so on. Can you get some help? I don't know, you see, biology is a different field, but you could probably...
That's a good question, but I think it's the particles are very different. In those cases, you're looking, the signals that are coming off of the particles are very, very different. For the case of biological particles, you usually have to either tag them with a fluorescent molecule that gives off light in a certain wavelength when excited, or you are looking at the intrinsic properties of intrinsic electrical or optical properties of the molecule. The goal of detecting very few biological particles in a mix of lots of other noise is really the goal here. And I think if, we can learn from other fields, it would be great.
No, in fact, when they have these emulsions, they have in one go before them millions of these particles, and then they try to find out which one is there through their experience. So similarly, if you have cells at one time, millions of them, photographs and so on, probably this is, I don't know, this is just because these days are the multidisciplinary.
I think imaging is a way to do that. Actually, I'm working with a colleague here who.
Can I interrupt you?
Yes, please.
Okay, thank you.
We are extremely thankful to you for your time and such a wonderful lecture and the information which you provided us. And we really feel proud that you are Pakistani origin and you are doing such a wonderful work abroad. So on behalf of all the audience and the organizer, I'm extremely grateful to you for your participation, and thank you very much for such a wonderful lecture. Thank you so much, and let us thank Dr. Rashid Bashir.
Thank you. Thank you. Appreciate it.
Ladies and gentlemen, after a very wonderful lecture by Professor Rashid Bashir from USA, this is my pleasure to invite Professor Dr. Najmul Haq. He's a Director, Office of Research, Innovation and Commercialization, and he's also Chairman, Plagiarism Standing Committee at Bahauddin Zikriya University. He's a Professor of Analytical Chemistry, and some keywords from his research, he's working on proteomics, lipoproteomics, phosphoproteomics. He's the expert researcher in the field of nanotechnology, and he has published more than 130 papers and 3 patents, and very highly cited, more than 3,000 citations. Please welcome Professor Najmul Haq.
Bismillahirrahmanirrahim. First of all, thanks to the organizers, His Excellency Professor Dr. Iqbal Chaudhry, Dr. Irshad, Dr. Reza, Dr. Yar, and all the audience. Before I start, I am starting 10 minutes late, and I am told that I have to finish in time. So I will be really fast and that would not be my fault. Please hold your chairs. Before I start, one comment. You were asking about the chips in the body. You should follow Elon Musk. He is working on one of the project that is Neuralink. That is chips to be inserted in the body for the diagnostic as well as for the theranostics, for the therapy purpose. And then there was a question about magnetic beads. I would be talking a lot about these beads created in-house. So what you saw in the previous lecture, all these technologies, they are shown to you, but at the back end, there is a lot of work which is done actually to make that possible. And that possibility is actually through the sample handling. That is how you handle your sample for creating any technology. Either it is bead-based or it is even chip-based. And the focus of all this lecture would be how can we target the signaling molecules out of the body, because the biomolecules present in our body, they are actually ready to speak to us. The need is we have to listen them. We have to targetedly find them first and then analyze and then get some useful information. And nanotechnology is one of the very good tools to deal with these complex samples. Now, what is the challenge? You can see in this slide, in the life science research, the omics sciences, genomics, proteomics, metabolomics, they all are generating a huge bulk of sample. And that sample, a compound sample, needs to be first separated and then to be analyzed. Starting from the genome, you can see there are approximately 30,000 genes. One gene on average produces 3 proteins. And then there are PTMs, post-translational modifications. And that number of protein becomes huge, almost 1 million, even more than that. And then there are so many metabolic pathways, they are producing the metabolites in our body. So all these things, they are present in our blood. So you have to separate them and then to characterize them for therapeutic purpose as well as the prognosis, diagnosis. And here I want to stress, early diagnosis is very important. Most of the diseases, they become almost incurable when it is not diagnosed proper time. Even fever, if it gets out of control, one will die. And cancer, diagnosed at the right time, will give you certain solutions. So earlier diagnosis is very important. And when we say earlier diagnosis, how much early? Even a single trace of biomolecule, even a protein, when it is released into our body, if you are able to characterize it, you have the information to deal with the disease. Now, as I said, you have to first separate these biomolecules. That means out of the blood, you have to first enrich it. And when you are doing the enrichments, this enrichment is not easy because the sample what you are dealing, I used to say the most complex sample in the universe is your blood. If there is anybody sitting here can challenge this, I really doubt it. The most complex sample in the universe is your blood. It creates such kind of information for you. It is actually the treasure hunt which you have to do. So these are the challenges what are written in the form of enrichment. The challenges are the dynamic complexity of the sample. It does not stay same. Even time to time, individual to individual, it is very hard to make a point that you can have some static information out the problem. And then the PTMs, post-translational modification of the proteins. Proteins, for its function, gets modified and then it starts functioning. At one time, there is a protein which is phosphoprotein. At another time, it will be glycoprotein. At another time, the same protein will be lipoprotein. So all these molecules, they are the signaling ones and they are controlling our processes. Because what is a disease? That is the malfunctioning of the right process. And why is this malfunctioning? Because the structure of the protein, because these are the biomolecules which perform function in our body, the cellular processes performed by the bodies, by the proteins. When its structure changes, its function changes. So you have to find out its structure. And my title of the talk was about this. We were talking about the beads. Now these beads which are shown here, this is enrichment media, the solid phase media. These are all the nanoparticles. Range of materials are available, material with different characteristics, and then there is a lot of chemistry done on this. But the most important thing, when you target any analyte onto the enrichment media, the results depend a lot on the sample preparation, how you deal with the sample. And these are very basic things, how you create these all the buffer systems. Because when you deal with biological sample, buffer, the change in pH is very important. It is the key to design a process. Does not matter if it is a chip, if it is a bead, anything. So this is very important how you deal with this, because this is existing, these are existing, this you have to create. And why we are focusing on the nanomaterials, the results have proven that they have got huge surface area, because when you are dealing with the complex sample, the concentration and the number of the biomolecules, they are huge in number. So you need huge surface area, and that surface area has made possible that only a trace quantity of nanomaterials, just 2 to 3 milligram of the material is enough to deal with the complex samples like serum and urine. And then selectivity. What is selectivity? You are targeting your analyte and how much your process is selective. Ratio is 1:11,000, means if you have 11,000 untargeted molecule, there is one which is to be targeted. The nanomaterials, by all these modifications, is able to enrich that, 1 out of 11,000. This is the highest selectivity ever reported in the literature in terms of the separation science. Then sensitivity of your targeted biomolecule to the level of 1 attomole. And these are the range of the biomolecules which you can work. Every molecule is a signaling molecule when it performs any malfunction. And recently we have switched to, because a lot of things were said about the cells, circulating tumor cells in the blood. That can also be one of the sample. Much easier than these kind of things to cater, CTCs, the circulating tumor cells. And recently we are developing a method on the basis that we can diagnose the cancer. These are the nanomaterials in their different forms. Like we can have metal nanoparticles, metal oxides, MOFs, polymers, etc. All of them we have created in-house, and they are used in any kind of targeting the biomolecules. I don't have much time that I go into detail, but these are obvious. The persons who are working in the nanotechnology field, they can very easily understand this is a range of nanoparticles or the nanomaterials available. As I said, class can be metal oxide, carbon based, because carbon family is very important. When we used to study in our O-level, A-level or metric classes, there were only 2 allotropes, diamond and graphite. And now a range of carbon family is available, and particularly the graphene in the last 1 decade, a lot of work has been done on that material. Then MOFs, huge surface area, huge porosity, metal organic frameworks. Then silica and polymers. Polymers are important because polymers, you can tune the properties as per your wish. You can design your own monomer and polymer is ready. And then they are functionalized in these forms. Like if you functionalize nanoparticle with bronic acid, it is good for glycoproteins. Helix is good for glycoproteins. And then MOAC, metal oxide affinity chromatography, immobilized metal ion affinity chromatography, reversed phase, normal phase, etc. And these are the characteristics. And this is also important because I will be just talking about it. The tool what you use after the enrichment, that is the mass spectrometry. This is again one of the most sensitive technique. In mass spectrometry, the salt tolerance, as our bodies, as our sample contain a lot of salt, the ions. So you want to get rid of these ions. The materials, they are also able to carry out the desalting from the sample. The ions are removed so that you get the ions in the mass spectrometry, which are not the adducts. And I was talking about the biomolecules. This is the huge range available in our body. These are the PTMs, the phosphoproteins, glycoproteins, lipoproteins, carbonylated, ubiquitinated, metalloprotein, ribosylated metabolites, phospholipids, glycospingolipids. These are all the range of biomolecules. And we have worked almost on the 90% of these, where we have enriched and we have tried to relate this with the cancer diagnosis. And I was talking about the technique MALDI-MS for the beginners, how it works. There is a MALDI target. Onto that, you put your analyte, you put the matrix, crystallization, shoot with the laser, and then you have the analyzer and the mass of those analytes. And importantly, this is a soft ionization method. Means you don't get any fragmentation after this. Almost no fragmentation, and that is the key, because biomolecules, we cannot afford to fragment them because the fragments produced, they are hard to analyze, they are huge in number. It is good for the organic molecules where mass is 100 or even 1000 Dalton, but here proteins, they are ranging to 300,000, 500,000 Daltons. So fragmentation is to be avoided. And then this is simple comparison. If you take a control and you compare with the cancer sample, you will get the patterns. And these patterns of the serum, they should be compared among the healthy and the diseased persons, and you can get those biomarker proteins. And now we coupled this material science with the MALDI, and we patented the technology named as MELDI. Now, how MELDI works, there is a small modification onto the MALDI target. There is an affinity surface to targetedly analyze the samples. Now, you have a complex sample with the affinity, you will bind the targeted analytes. The rest of them is washed away. You put the matrix and then you analyze it. And now you have the MS, or the masses, resolution, signal to noise ratio, etc. of the targeted analytes. But very important thing, when you change the material, it's not only the chemistry which is important. This is very important point to stress. These four materials are listed in front of you: cellulose, silica, diamond, a polymer. A same kind of functionalization, same chemistry has been done on different materials. From the same sample, under the same conditions, every material produces different results. And that is actually the problem. It's not only the dynamic complexity of the sample, but even materials, they produce different results. And the reason being, every material has got its own characteristic. That is surface area, porosity, etc. So few of them will allow lesser chemistry, few of them will allow more chemistry. Chemistry means when you do the functionalization, the number of functionalized groups which you get onto the material, they are different from material to material based on the surface area. And as a result, every material produces different results. And you can see four spectrum, they are compared, and there are huge differences, having same chemistry, having same enrichment method, but giving you the different results. This is the same continuity of that. And this is what I was talking about, the pattern algorithm analysis, the machine learning, the artificial intelligence. These all processes these days, they are converted into certain kind of algorithms. What is a machine learning? Machine is actually given lot of data so that it can develop certain sense to give us the conclusive evidence of diagnosis and prognosis. This is one of the study where gallbladder carcinoma was studied by the in-house material, that is diamond, immobilized with metal ions loaded with iron, and you get different kind of reported biomarkers which are already existing in the literature. And these are the validated biomarkers, the proof of concept. Why should we work on it? Because whenever we plan a study, there should be proof of concept, that is on to which you are hypothesizing, you are making your hypothesis. Is it the right hypothesis? Among the literature, lot of phosphoproteins, they are linked with different kind of cancers. You can see these are the FDA approved biomarkers from prostate, breast, cardiovascular, etc. These are the phosphoproteins present in our body. So you have to enrich them, and then you have to couple it for the diagnosis purpose. As I said, you can use the metal oxides and the metal nanoparticles. One of the tools to enrich them is immobilized metal ion affinity chromatography, and other is the metal oxide affinity chromatography. This is much easier because you can create a range of metal oxides from all the periodic table, the metals. You can create their metal nanoparticles as well as their ions as well as their oxides for doing these enrichments. Now, when you do the enrichment, you have just seen Professor Rashid, he was talking about that the process is taking 4 to 5 hours. And that time has to be reduced actually for the analysis purpose. And how you do that, you actually transform your technology, how you do the enrichments. Beads can be used in multiple forms. Keep in mind, these all the processes, the 3 main processes mentioned here, they are using the beads. Now, beads used as SPE material in biological vial, in beaker, in test tube. Beads used on the target, means on the chip, on the surface, and then beads used in the tip, biological tip, that is micropipettes having the disposable tips. You can create that. And I will show you at the end, the process of 2 to 3 hours, 4 to 5 hours can be brought even into the matter of minutes. And how many minutes? Only 2 to 3 minutes. Even the commercial technologies, they are consuming 2 to 3 hours. And the reason being, the process takes a lot of time. You have to make the smarter strategies, and TIP is the method which is sensitive as well, which is selective as well, and more importantly, it gives you all the results in 2 to 3 minutes. And this is the tip which is shown in front of you. The bed height is shown, that is again less than 1 centimeter. That all the separation media of 2 to 3 milligram that has been brought into the tip, and separation is very effective. This is only the bed height. You can see this one. All this area is empty. And this tip is made up of lanthanum oxide. You can also do the solid phase extraction in these biological vial, and then I was talking about the magnetic beads. These are the magnetic beads. You don't have to do the laborious centrifugations, filtrations, etc. What you do is you do the enrichment and you do the magnet. Just put the magnet here. What will happen? All the beads will get attached to this magnet, and supernatant can be discarded. Otherwise, why it is taking time? It is taking time because you need centrifugation. And if your surface area is high for the nanoparticles, its density will be less. And if its density is less, you need more time for the centrifugation. So that is the reason it is taking a lot of time. If you make your beads magnetic, time is reduced. And then you can do the on-target enrichment. That is, you have the multiplate. Onto that, you do the chemistry and you do all the phenomena on the surface. And I was talking about the sample preparation conditions, what you have to optimize. Like here, you can see when you do the targeted separations, you bind your targeted analyte, but the untargeted ones, you have to wash them. So how you wash them? You have to keep your targeted molecule bound to the surface and remaining content is washed. So eluting buffers, washing buffers, they are very important. They need huge optimization at the back end. The chips, every technology is not going to work till you optimize all these conditions. And they have been optimized, and you can see here, what is the difference? 80% citronitrile in 0.2% trifluoroacetic acid, and these are different, and results are also different. Washing conditions, elution conditions, I'm not going into detail, but these conditions are very important. And then you can also see the difference As I said, range of metal ions, they are available. They can be immobilized onto the surface. The same material, when you vary the metal ions, remember from our FSC, the shape concept, soft, hard, acid, and bases. And every metal is different in that regard. And the biomolecules, they have also got their own isoelectric points. Keep in mind, isoelectric points, and they will definitely respond to these metal ions. These are the basic chemistry elements, but if you vary them, you will get a different kind of result. And when you use the material in the individual form and then you make the composite, results will also vary. Like here in this case, cellulose, silica, and when you make a composite and then you load with the metal ion, you will get different results. As I said, the selectivity study for 1 to 11,000, and that was obtained from this material, gadolinium oxide. As I said, this is the highest ever selectivity reported in the field of separation science, and this is the proof of concept given here. You can see here, this is a beta casein. Out of that, phosphopeptides has been enriched, and the ratio made is 1 to 11,000. This is the chemistry of gadolinium oxide, how it works, and then the sensitivity of the attomole. Again, gadolinium oxide worked till the range of 1 attomole. 1 attomole means if your targeted analyte is present in this concentration, your method, the optimized method, it is not only the bead, your sample preparation conditions as well, they will enable to enrich to the level of 1 attomole. And the biomarkers, the signaling molecules, keep in mind, they are not in the millimolar or the micromolar concentration in your body. They are present in femtomolar. They are reported to be present in the femtomolar concentration. And why I am stressing on this selectivity and sensitivity, that your method should be that much sensitive that it can reach to that layer. Otherwise, our body contains abundant proteins. They are present. Out of those abundant molecules, you have to target this lower concentration range. And when these all methods have been applied to the standards like milk casein, huge enrichments, these phosphorylations, they are how this phosphorylation happens, that is the serine amino acid, etc. I will not go into detail. Optimization have been done in egg yolk. And then, very important, you can also do the multiple targeted in one experiment. Like there is one class of molecule that is the phospholipids and other is the phosphopeptide. You can enrich them in one step. In one step, you will targetedly bind phospholipids as well as the phosphopeptide. But in the elution method, in the successive elution, first you can elute the phospholipids and then the phosphopeptides. And then phosphorylation is related with the cancer. This data is available on the website of National Cancer Institute, how this phosphorylation, only I'm talking about one of the PTM, how that can give you the information about cancers. These are all the enrichments. Then you can study the peaks. These peaks are already reported in literature. They are relevant. These are the phosphopeptides which are relevant to the cancers. And keep in mind, it's not the phosphopeptide which are present in your body. Actually, it is the phosphoprotein. But when you analyze them, you don't do it in the form of phosphoprotein. Rather, you first digest the sample. You bring that bigger molecule of protein into the peptide form, and then you enrich it to get the information. Because a complete structure of protein is hard to analyze, so you do the small digestion first. This is all the proof of concept, how these phosphorylated molecules, they are related to the cancer. I will wait till you make the picture so that, okay, fine. And this is the clinical importance of phosphoproteins, proved through mass spectrometry. And then the method is also able to detect the rare phosphorylations, which are really less in number in our body, but the method is too sensitive, too selective, that even it can analyze the rare phosphorylations at serine and at threonine. It is proved with the masses. These are all the rare phosphorylations. And then you can link the information from genome to proteome and proteome to genome. This is just the linkage of the two. This is very important slide where you are getting the number of positions already present in the cancer types, how many they are. They have been quantified regarding the cancers. And was the philosophy what I have discussed till now, that is bind some, elute some, and identify all. That means you will bind the targeted analytes and then elute them, but never ever try to throw any kind of sample, even if it is the supernatant, it is the eluent, because it has been separated for the purpose. It's not that only phosphoproteins are important. When you are targetedly binding the phosphoproteins, there are glyco, there are lipo, there are many contents. What you should try, ideally, every kind of biomolecule should be studied. So it is fraction by fraction. So you do one of the fractionation, and then the remaining sample should be dealt for another kind of fractionation, so that you don't lose any information, because the signaling molecules, they are huge in number. They belong to more or less every class. After the phosphoproteins, we can talk about the glycoproteins, the glycopeptides. There are different type of glycosylations in our body. Yesterday, Professor Raza was also talking about the glycoproteins. They are also very informative. They are huge in number in our body, and there are multiple type of glycosylations. We are interested with N and O glycosylations particularly. And what are the enrichment methods available? There are many which can be utilized, but we are interested with boronic acid chemistry and the Helix. Helix is hydrophilic interaction liquid chromatography. By that, you can do the enrichments. And this is the chemistry what we have done. What is Helix? What are the hydrophilic substances? Very short way to describe it, that make all the polar content available onto the material as much as possible. You can see here all the functionalization have been done where you have the charge separation. Material has been made polar because the polar content is hydrophilic in nature, and hydrophilic substances will bind more importantly with the glyco stuff. And I can see time is getting nearing. I will not go into detail. These are the different Helix Sorbents which are produced. Even in the tip, we have created material on the basis of piperazine. These are the materials well characterized, their adsorption capacities, etc. They have been calculated. Very important. These are actually the main aim of a separation scientist when you get such kind of uniform beads. And again, this material 1,2-epoxy 5-hexene DVBA loaded with aminodiacetic acid. This is the highest ever surface area reported, 433 square meter per gram for such kind of materials. It was very less dense, but very uniformly populated. These are different materials. I'm not going to detail. These are the optimizations. And this is very important slide. When you do the blood serum analysis, you can see the number of identified glycoprotein. This is one material, this is another material. In this start, I was talking about this. This material enriching 113, this is enriching 129, and 203 are the common glycoproteins. So when material changes, you can see there is again a huge diversity. And in this case, these are 1, 2, and 3 materials. You can see the common things and still there are different. So by changing the material, the enriched content is also changing.
Could you please conclude?
Yeah.
The next partner speaker, sir.
Lipoproteins, metabolomics. And this I can't skip. That is the acknowledgement. My whole research group, my professor in Austria. I should appreciate him a lot because I learned all the separation art from him, all the institutions who have worked in collaboration. University of Karachi as well, because in the start, in the beginning, we were using the multi-MS there, but now I think it's not working. When it will working, this will be reflected again here. Thank you.
Thank you very much, Professor Najm. We are getting late, so I will take just one very quick burning question from the audience. Yes, please.
And we can discuss in the tea
Yeah,
if you
in the
will have
tea,
other
yes.
questions, because we have to cooperate. Yeah.
Possibility of the various types of the mixed oxide. So what may be the chemistry behind?
No, what was the possibility? Please repeat the question.
During the synthesis of the gadolinium oxide, there is a possibility of the formation of the mixed type of the oxide, like GD2O3 gadolinium oxide. So what may be the chemistry behind this, and what may be the role?
That's
of
very, very important question. And in the separation science, it is always asked to me that where is the characterization, how you do the purification of the material. And the good thing is we never do that. Gadolinium oxide does not matter how many isoforms are produced. We are still interested with the sample preparation optimization. If you do the purification, it is a hard job. Like when you make a composite, this whole composite is never uniform. Somewhere you will have one kind of metal oxide particle, otherwhere some, till you make a hybrid. So the purity we never worked on. Sure, it should be worked, and then it should be seen that how it is affecting the results.
So do you think that we can increase the selectivity by purifying?
Yeah, that's what I said. It should be checked, and maybe it is getting improved.
Thank you so much.
Thank you very much, Professor Najm. Ladies and gentlemen, please join me to thank Professor Najm. Wonderful job. So that's a very short break for the tea. We have that's very exciting lecture. We will please come back here by 11:25. Very short break, and we will start next session sharp at 11:25, please.
And then you can start your lecture.
Yeah, thank you very much.
It's again my pleasure to introduce Dr. Mohammed Ashraf Gandal, who is distinguished professor at Physics Department, coordinator of Laser Research Group at King Fahd University of Petroleum and Minerals. He is senior research fellow and he holds many publications and positions. His research interests are in the areas of nanotechnology, photonics, quantum optics, material science, energy harvesting, energy storage, sensors, laser remote sensing, environmental pollution monitoring, and many more. Dr. Gandal has published more than 550 research articles in leading international journal of the world. He has 30 US patent to his credit. His H index is more than 50. And amazingly, his citation is like more than 9,000. He is also member of many international societies and is on the board of various international journals. So, Professor Ashraf Gandal, we are looking forward for your lecture, and floor is over to you.
Bismillah ar-Rahman ar-Rahim. Nahmaduhu nusalli ala Rasul al-Kareem. It is my pleasure to be, and I am thankful to the organizing committee, especially Dr. Shad and all, and Dr. Jar is my friend. He worked here in my university. He knows my university very well. I'm going to talk, actually a lot of talks was done today and yesterday on sensing of cancer cells and other diseases using nanotechnology, but I will just talk briefly on how nanotechnology can help us to cure the cancer and what are new modalities and new treatments coming into the market using nanoparticles and nanomedicines. This work is done at my university on the left side. This is my campus. On the right side is my institute where I work and where we do this work. The outline of my talk is that I will just give you a brief introduction and the motivation behind this work, what is cancer and what are the present technologies or the techniques which to cure the cancer, although this is not a curable disease still. It has a lot of problem. It's very costly disease. And I will talk about a little bit about how we synthesize nanocatalysts. There was a lot of talks gone on wet chemistry method, but I will introduce a new method which is based on lasers to generate nanoparticles of any size, starting from quantum dots to large, particles and nanocomposites. Then I will present three kinds of studies which we did using three kinds of, catalysts. One is the silver-loaded W3 nanoparticle to cure HeLa cancer cells, and then cervical cancer cell using a nanocomposite of W3 as ZNO. And then I will talk about waterborne pathogen E. coli, which is mostly in your urinary tract infection. It causes E. coli bacteria, how we can get rid of using nanoparticle. And then I will conclude my talk. The threats to our ailing humanity is that there are many diseases which we are facing now. Some are rich country diseases like diabetes and hypertension, but it's coming into our, due to our lifestyle in Pakistan, it's spreading very fast. I will focus on cancer, and we all know that the cancer is the most killing disease. I have data from the last year that it killed almost 9.9 million people worldwide. More than 30 to 50 million people are already have got the cancer, which is still not cured. So what is cancer? Cancer is that in normal tissue, in a normal human being, we have a balance of the healthy cells and the cancer cell. But when this balance is disrupted, then there is uncontrolled cell growth due to the loss of cell ability to undergo apoptosis. Okay, apoptosis is that by nature, there is a self-destruction of any cell which is against the healthy cell is killed. But if this balance is disrupted, then the cancer cell, they grow very fast, they mutate, they can transport from one organ to another organ of the human body. And it is a major public health problem worldwide. And the most killer, almost 9.9 million deaths happened last year. And it has superseded now the heart diseases also in everywhere. The only solution at present, there are three major treatments which are available: surgery, radiotherapy, and chemotherapy. We all know that radiotherapy and chemotherapy has a lot of side effects. So a person goes for a cancer treatment, then he lose a lot of other organ due to these side effects. We all know that. And it is a very costly disease. Therefore, new methods which are safe, which has less side effects, which cannot damage the normal tissues, so they are in demand. And nanotechnology has the potential to play some role in such achievement to achieve these goals. What is nano? Nanomaterial, all we know. Just for my students, I just like to teach them that the surface to area, surface to volume ratio can be increased. For example, if I have a cube of 2 centimeter dimension, my surface to volume ratio is 3:1. But if we cut the same cube into 3 nanometer, you can see I can have surface area to volume ratio 6 billion to 1. So that means for 6 billion reaction can take place when I go to a nanoparticle material, or we have 6 billion engine working for us to do some work for us to act as for any reaction. So that's the big advantage to have a nanomaterial. And we know that when we have a nanomaterial, its optical, physical, chemical properties change. Just you can see from centuries, we know the gold is yellow, but now we can make a gold of any color just by changing the size of the gold. And here you can see just changing the size of nano quantum dots, which we have produced in our lab. You can just change the size from 2 nano to 8 nano. You can get the size, color is completely changing. So we can control the optical property, we can change the chemical property, we can change the physical property. So there are many, one can use this. So the therapy which I'm going to introduce is called chemodynamic therapy for cancer treatment. There are many therapies available, but chemodynamic therapy is basically based on reactive oxygen species generation, ROS. And this can be done in many ways. And most of the easy ways, Fenton type like reaction, which you all chemists know very well. But we will do here basically the generation of these hydroxyl radical OH, which will be very effective in killing the cancer cells we generate by just photocatalysis process. When we talk photocatalysis process, photo mean light, catalysis, a catalyst is active by the light, we can generate these radical very easily. And this is shown in this just animation. You can say we have a semiconductor. We know semiconductor has a valence band and a conduction band. And when we shine the light of a proper wavelength or proper energy above this band gap energy, 3.2 electron volt, for example, for TiO2, we can generate electron and hole pair. So the electron are excited to the conduction band, and we have holes down. And this upper side become as a reduction site, and the lower side become as an oxidation site. So we can generate in the water, we can break the water molecule into OH radical, or we can generate very highly excited oxygen. And when these two are highly excited oxygen are these two radical, they attack the cancer cell. We can kill the cancer cell, and mostly because this is organic matter, so we can generate carbon dioxide and water, which are safe products. So this is how the photocatalysis works. And my catalyst, what I am trying is, we have loaded silver on WO3 nanoparticle, and we use this as for chemodynamic therapy. Why we need silver here? Because the band gap of WO3 is 2.8 electron volt. And also there is another challenge that these electrons which are excited into conduction band, they will jump back so they can recombine, which I don't like. That I want to use these electrons to break the water molecule to generate OH radical. So what I use is that I can use this silver doping on the WO3. So now by, this surface plasma resonance enhancement, I can now use the visible light, and then I can bring the electron from silver to the conduction band of this WO3, and I can generate these radicals. And those radicals then attack on the HeLa cancer cell. Why we call this HeLa? This was a African-American lady whose name was Hynette Lacks, and she has a mother of 5 children, and these cells are always used as a culture lines because they can grow in any media, while many the cells of cancer, they cannot be used. Now I will go to the techniques which we use to synthesize nanomaterial. And there are 2 ways we know: bottom-up approach, that we start with atoms and molecule and we build the nanoparticles. On the left-hand side, you can see this is done by wet chemistry. It has certain drawbacks, it has certain advantages. What Dr. Shah yesterday told us, that he can control also through wet chemistry the size of these and to even quantum dots. But it has also that you can generate a lot of chemical waste. We have developed recently a laser-based technique which is called top-down approach. Like in centuries in India, we will do this silver films of to protect our sweets, for example. That was a ball milling, okay, and we used as a ball milling technique using pulse laser ablation. I will just explain you what is this technique in next 2-3 slides. Normally, whenever you shine a very high fluence laser, we can ablate the material because we generate here 10 to 5 degree Kelvin temperature, and we create this a lot of material we ablate. And this material, if it is in a liquid environment, we can create with the laser fluence a plasma on the surface of this material, which is a bulk material, which we like to convert as a nano. We can create a cavitation bubble in there, and that when cavitation bubble explode, it can create a shock wave, and that press the bulk material, and it makes the nucleation process, as well as it will make the nanoparticle by that high pressure wave. The setup is just very simple. We have a laser, and by just controlling the laser parameter like laser wavelength, laser energy, laser pulse duration, and the medium, liquid medium, or we add some surfactants if we like. But without surfactants, we can even control through the size. So here, this is one example that you have cadmium selenide, which is black, but when it becomes a quantum dot, the color completely changed to yellow. This is the setup that we have a neodymium YAG laser, we have a turning mirror, we have a focusing lens here, and we have just a magnetic stirrer. It's a very simple technique and very simple setup, but a beauty is that within 15 minutes, I can generate a nanoparticle of the size which I like just by controlling the parameter, while chemical methods sometimes take 3 days, 4 days or more. So it is very costly, very rapid, and a green technique because we don't use any precursor here, anything required. Just by change, we can do a lot of outcomes. For example, we can formate new chemical bond. We can break chemical bond by laser. We can do the phase transformation of these crystal, which we actually synthesize, or we can resize the nanoparticle from bulk particle, just controlling the laser parameter. And this is one of the example which we have. You can see a nanocomposite Ti2, ZNO, CDS, and you can see the band gap. We can change CDS as 2.4, which is in the visible region, while zinc oxide is 3.7 in the ultraviolet, TiO2 is 3.2, also ultraviolet. So when we make this nanocomposite, we can really bring this nanocomposite to the visible light region very easily, and we can make many applications for water treatment or for other application using sunlight. Now I come to the studies which we done. First study we done with the silver loaded W3 nanoparticle. Here we use the wet chemistry method, sodium, tungstate, we use hydrochloric acid, silver nitrate. When we mix through precipitate process, we can get a very, silver dot W3 nanoparticle, which we characterize from the normal and high resolution transmission electron microscope. On the left-hand side, you can see that the particles are mostly spherical shape, some of them. Okay, and we can also use the crystal lattice also very much through this selected area electron diffraction technique. So you can see very well that the particles of the nano size, and we also through field emission scanning electron microscopy and EDX technique, we can see that make proof that there is a W3, there is a silver W3, you can see all these peaks and their percentage in the nanocomposite. Also, we have verified through XPS analysis technique that whatever material we produce, it contains silver or it contains W or it contains oxygen. So you can see clearly the peaks by the binding energy, what we measure through XPS technique. Since we are interested to go to the visible region, so band gap determination is very important. You can see that the actual band gap when we have a pure W3 is more than 2.8, which is around 420 nanometer. And when we get 3% silver dot, you can reduce to 2.6. So you are going toward the redshift to the visible region. How we detect our HeLa cancer cell, whether they are treated. So we use basically fluorescence technique. So we use LMR blue in these wells. There is a cancer cell, we put our catalyst, we put LMR blue here. And when this HeLa cancer cell, they react with this LMR blue, they become red. So from this fluorescence intensity, basically we measure from this the plate reader that how much HeLa cancer cell has been died through this. So these are the results which we achieved. You can see here down when we have 3% silver W3 concentration, the cell viability goes to almost, almost 95% killing is there very clearly. You can see the effect here. And this work we recently just published in Nature Scientific Report. This is where we have plotted cell viability versus the concentration of our catalyst. What you put, we have to put just very small amount, 200 microgram per milliliter. And you can see that at this corner on the right corner, that almost more than, 95, 96%, the HeLa cancer cell has died. This is the plates which we studied through that, and the color will tell us that how this HeLa cancer cell has left behind from the fluorescence intensity. Now I go to the second experiment which we used here, W3 ZNO nanocomposite. The main challenge, as I told you, that W3 absorb around 420 around nanometer. We like to bring this catalyst to the visible region. So we have prepared W3 ZNO nanocomposite. Again, the excitation process here, we have a heterojunction between this and this two catalysts, and we can shine, first of all, for example, a 2.88 electron volt to this, and then we tunnel the electron to this. So we have both way, we shine the light. We can use either laser light or we can use a broad spectral lamp, and we can excite this and we can generate OH radical, and these OH radical then goes and target the HeLa cancer cells, and then the HeLa cancer cells are dead by these radical reaction. The synthesis is just a flow chart here. You can see we start again with sodium tungstate and ZNO in deionized water, and we put hydrochloric acid, and finally we get the very beautiful, nanocomposite ZNO-WO3. We do the morphology study using HRTEM and SAD analysis, and we see the crystal, these plane and the lattice, 101, 100, 002, and you can see that the particle are of the order of around 25 nanometer to 50 nanometer here. And we do also SEM analysis, and we also study the band gap using UV diffuse reflectance technique. The important thing is that how we can monitor that this electron, which is excited to the conduction band, jump back to the valence band. And that we can do with the photoluminescence analysis very easily. If the emitted light is more, that means the electron hole recombination is taking place. And that is clearly, you see, when we put ZNO, this red, you can see there is a lot of emission, emission intensity of photoluminescence. That means there's a pure W3 is not good for our work, because whenever electron, we bring it up maybe 50% or more, jumping back to the valence band, and the light is emitted. But when we put W3 ZNO, you can see the emission has decreased a lot. That we are controlling basically by doping ZNO to W3 to prepare this nanocomposite. Then we are controlling also electron hole recombination process. And by this way, the efficiency of our catalyst will enhance definitely. And this is the final results. You can see here again that we have almost 94% killing of these cervical cancer HeLa cells with this technique after 60-minute exposure of the light on the catalyst. We can also study the cell viability, and you can see the best result. This is a blue curve. You can see with the W3 ZNO, we can achieve very high, the percentage that the killing of the HeLa cancer cells by this technique. And this is, you can see 96 or 90% we have the viability. Now I go to the third experiment where we are working on killing of E. coli bacteria, which is very dangerous for our urinary tract infection. And we have also used here a catalyst, which is, you can say, cadmium selenide and graphitic carbon nitride is loaded on that. And we can see here clearly that how our excitation scheme they work. So we have CDS on the right side, and we have a graphitic carbon nitride. So CDS has 2.4 electron volt band gap in the visible region, while this is close to the UV region. So when we generate these radical O2 dot minus or OH radical, it attacks the bacteria membrane. Bacteria is your organic matter. So the bacteria is completely deactivated, which I can show you later on the results. The preparation here we use basically the wet chemistry as well as the lasers. So first we make bulk, from melamine, graphitic carbon nitride particle and CDS particle. Then we mix them on a magnetic stirrer and we break second harmonic, which is a 532 nanometer laser. We focus on this and finally we get a very beautiful, nano composite CDS at graphitic carbon nitride. And this work recently we published in Collide and Surfaces. You can see here different color before pulse laser ablation. The color of your tungsten oxide is this and CDS is this. And when we shine the laser, after the laser, you can see that down here on the right hand side that the color of both the particle have been changed when, although they are become nano, and after nano, even when we mix them under the laser, and we make a solution, the nano composite, the completely color and the property of this material has changed. And here we do the XRD to test that whether we have CDS, we have graphitic carbon nitride, and then you can see all these peaks. When we have this nano composite on the right hand side with XPS, we have proved that all these, carbon is there, nitrogen is there, CD is cadmium is there. So all this we can verify through our XPS machine. These are the, high resolution XPS, and we can also identify there atomic level. You can see S state, P state, D state, all these states, we can identify them. And then finally, we do this UV diffuse reflectance spectra through Kubalka-Mall function. We can calculate the band gap energy of this catalyst, what is we produce. So you can see the band gap has shifted toward the visible light because it has decreased. And we know that the energy is inversely proportional to the wavelength. So we reach to the visible light. This is a photoluminescent spectrum again that you can see we can control electron hole recombination. Pure graphitic carbon nitride you can see is green, while when we have this nano composite, the emission has increased almost more than 60%. So electron hole recombination is controlled very well by this nano composite. And these are the plates which you see that in the left you have control E. coli. And this is just pure graphitic carbon nitride. And when we have CDS and graphitic carbon nitride, almost we get very high killing of E. coli bacteria. And we can also calculate the reaction rate or a constant from there by plotting N over N0. N0 is your initial concentration of E. coli, and N is with the versus at different irradiation time. We can get very high radiation time. And the morphogenesis of E. coli bacteria before killing, you can see the bacteria is alive and now it is dead on the right hand side. Very clear indication that bacteria is dead now. In conclusion, we have proved to you and demonstrated you that laser-based techniques are really very useful, and within short time we can produce a nano composite starting and nano materials starting from quantum dots to any shape. We can make spherical, we can make nano wires, nano tubes, all just by controlling the laser parameter. Then we tested them for basically the killing of HeLa cancer cells using W3, AG.W3, and then ZNO-W3. And we proved that AG.W3 gave us better results than even the other, the catalyst. And we also proved that cadmium sulfide graphite carbon nitride is very effective in disinfection of E. coli bacteria. Uh, just to give you that we have ISA journal publication. We are not working, as Dr. Shah told you, just on photocatalysis and its application in different areas like water treatment, bacteria disinfection, and also conversion of CO2 into value-added hydrocarbon like methanol, for the controlling of the for carbon management. But we have a lot of work doing on energy harvesting and storage devices. We are working on third generation solar cells, and also we are working on supercapacitor hybrid type, where we can use light to generate electron and then we store them. We are using perovskite material also, and based on that, we have many. Graduate student, we have 25 theses so far done and 30 master student, 40 projects we completed. Almost 24 patent has been already licensed to us by US Patent, 20 are pending under review. And anybody who is interested in our publication, you can go to scholargoogle.com under my name, so you can see them, the publication under that. I think I have a problem with my slide shift. Just I want to actually acknowledge that my last slide. Thank you very much for listening, and we have, I think, still 5 minutes if you have some questions. Hello? Hello?
Thank you very much for such a nice lectures and such an impressive CV. And we are really impressed the way you are working. So there is one short question, because Dr. Ashraf is about to leave for his class.
Yeah, please, you can welcome. If you have any question through chat, you can send me question. Inshallah, I will answer you. I have to acknowledge actually my students there. I have a multinationality group, starting from Yemen, India, Pakistan, Nigeria, Singapore, and some Chinese students who are with me also, but now they left. And these are the agencies who funded my different projects. King Abdullah City for Atomic Energy and Renewable Energy, Kakir. I'm a senior research fellow. My physics department, I have to thank Center of Nanotechnology and Deanship of Scientific Research. And CAXT is a funding agency here in Saudi Arabia. We have a project, and COUST as well. So we work with them. So these are the people who are behind this work. So I have to acknowledge this. I'm not the alone who worked this. This is the student work which I presented.
Thank you, Dr. Gandal, for a very nice and informative presentation. I am following your profile since we met back in 2016 at Comstech, Islamabad. My question is related to your anti-tumor studies. You have performed phenomenal studies in regarding the using different formulation. Why didn't you choose to go to the in vivo anti-tumor efficacy of your formulation? This is first part of the question. While the second part is that, why did you choose only the HeLa cells? Why don't you go to HS7 cells or some other MTI cells? You can see there are a lot of...
Really a great question, and I like that. Unfortunately, I am a physicist. I don't have the lab incubator and all this microbiology lab under my control. This is work which we did with actually I have a female student, Palestinian student. She came from Riyadh, don't have the facility, so I start work with this, and we did this work in some hospital, these studies. HeLa cancer cell, they are the culture line is the only those cells which you can grow and which you can feed, and they are the best for culture lines. That's why we selected HeLa cancer cell. That's the only thing. The other, your question is that why we don't try. It will work definitely on tumors, but we need a laboratory or a person who can collaborate with us. If there is anybody from Pakistan, I'm really willing. I can make my material and ship to you guys, and you can try in Pakistan.
Okay, thank you very much. We will be inshallah in touch with you regarding the anti-tumor formulation development. I am from Qaid-e-Azam University, an assistant professor in the Department of Pharmacy. Thank you very much for your.
By the way, one more point is that recently we study some herbal medicine. They really work a lot of our tumors, by the way. That is a partial study. For example, if you take the moringa seeds and you crush them and make them nano, they are really very, very effective in killing off these tumor cells, and they have very zero side effects because it's a natural product. So why not you try that, and we can support you in that research.
Okay, thank you very much, Dr. Ashraf.
Okay. Nice. This was great. I'm thankful again to the organizing committee, especially Dr. Rashid, Dr. Jar Mohammed, and you, Shah Sahab, for this, that you gave me this opportunity to talk about this. Thank you.
Thank
See
you.
then. Assalamu alaikum.
Professor Rahman, could you please hear us?
Yes, I'm ready.
Excellent. Ladies and gentlemen, next speaker with us is Professor Dr. Mohammed Basiaruddin Abdur Rahman. He is currently the Senior Professor of Chemistry and Dean Faculty of Sciences at University of Patra, Malaysia. He received his PhD in Catalysis Chemistry from University of Southampton and followed by a postdoctoral studies in Structural Biology from University of Edinburgh. He was appointed as distinguished visiting scholar at the University of California, Berkeley, and his research areas include biocatalysis, chemical biology, and computational chemistry. And he has published more than 200 papers, filed more than he has published, more than 200 papers and filed more than 30 patents, and graduated more than 100 postgraduate students. He is the fellow of Royal Society of Chemistry, UK, and International Institute of Advanced Materials, Sweden, and Academy of Sciences, Malaysia, and he's also the president of Malaysian Analytical Science Association. Ladies and gentlemen, join me to welcome Professor Rahman, please. The floor is yours, sir.
Thank you very much, Mr. Chairman. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi ta'ala barakatuh, and good morning or good afternoon over there, because here is already good afternoon. Yeah. First, I would like to say thank you very much to Comstech, particularly Professor Rashid for inviting me. It is such a pleasure to be here to listen to many very good work from the speakers before. Yeah, so I would like to start that how we evolved from doing something that totally different to what I'm doing or what I would like to present to here. So let me start with the University of Putra, Malaysia. Actually, we are not that far from Pakistan, and we basically the research university in Malaysia. And our focus is actually, or the niche area of UPM is agriculture and food security. But having said that, we also have veterinary medicine school, faculty of medicine, yeah. And one of the oldest faculty in UPM is the Faculty of Science itself. Currently, we are ranked in QS ranking the 132. Yep. Okay, so let me start with very simple how I evolved into what I've been doing right now. We are actually more into the catalysis and biocatalysis. This is how I've been trained. But then during the process, we did a lot of, in order to get a better enzyme, we did a lot of structural biology, the mutation, and at the same time we also immobilized. And this is when I start to get involved more into what we call as the nanomaterial later on. Other than that, my focus is still linger around the structural, computational, and chemical biology. I think this is an overall scope that how can we do the very interdisciplinary or transdisciplinary science. Yeah. So from there, we develop lots of, or basically we also file patent to a lots of our chemicals or products, particularly from palm oil. Yeah, so we have many high value added products. For example, what we like to show later on is our wax ester being patented in the United States and some of our MCG and also MCT. Other than that, we use it for the different application. And I also did mention just now that we also work on the immobilization, because I think this is how the idea we transfer from immobilizing what you call these macromolecules or enzymes then to the drugs. Other than using all the commercialized, and we modified many of our natural abundance like mica, feldspar, and choline, we also synthesize our own layered double hydroxide, even the ionic liquids, but we make it very, what you call these, very like kind of solidified so that we can coat the enzyme. And recently we start to work on the mesoporous silica nanoparticle, but we use ionic liquid as a template. And very recent, we start to focus on metal organic framework. So from there, this is how I would like to enter more into what we call as the nano delivery, or particularly nanomedicine for this workshop. In the beginning, we focus more on the cosmetic and cosmeceutical. Basically, we use for topical application. We also use for the pesticide, particularly herbicide and fungicide. And my colleague also working on the pharmaceutical, particularly for the blood-brain barrier, only in 2015, 2016, when we were invited to study for a consortium of Malaysian research, but that focusing on nanomaterial, and in particular for health. And this is when we start to look at the aerosol, or in delivery system, we call it as a pulmonary delivery. Other than that, we also focus on the computational colloids, because we have a huge amount of data that related to the preparation. This is more like kind of state of the art, but we need to make it a concrete science. So from there, the focus in nano emulsion is actually, of course, between 20 to 200 nanometer. But as I mentioned just now, we have data from the ternary phase diagram. So from this experimental data, we convert it into the computational. And from there, we can save a lot of chemicals, and also a lot of prediction have been made in order to explain certain behavior of these colloids. So we are not limited to using just molecular dynamics. We also use coarse grain and Monte Carlo simulation, because in certain cases, the system are too big to be studied. Right. So this is actually way back in the 2010 and below, where we focus in the beginning. For example, this is one of the most studied, the prediction by Davis way back in 1994, in order for us to understand how the ternary phase diagram between the surfactant, oil, and water give a certain kind of a shape and size for particular composition. So from here, our computational prediction being able to match our experimental data, where certain composition will give you like kind of a lamella, ellipsoidal, or even what you call this a spherical. So it is very important then we use this size, shape, and the structure of my cell for our prediction. For example, in this one, we put the ibuprofen, and what we found from our experimental data is actually a fast release. And from the computational work, we saw that the ibuprofen basically at the outer layer. That's why it's suggesting the instant release of the drug. And other than that, we can also look at the self-assembly of the PKOE, which is actually our palm kernel oil ester that we use as the like kind of a secret ingredient for most of our formulation. Here are some other, what you call these, just a moment. Okay, here are some of our other formulation that we studied. In particular, we use a complete system because just now is a very simple system, the one that I showed you previously. But in this one, we studied with the MD simulation and then coarse-grain MD simulation for the ibuprofen, and same as well for this case, what we use for the DPPC, Cremofor, and diclofenac. We can now understand better and how to relate between the fast release, sustained release with the composition of the drugs. Yeah, but at the moment, we also really look forward to understand in terms of the experimental, in particular, the actual concentration of each nanoparticle inside the system. Yeah. So let me start with the actual work that I would like to share with you. I'm so relieved that the previous presenter today and yesterday been talking a lot about the nanoparticles, so I'm not going to elaborate more on that. And also previously, we also Professor Gandal talked about the cancer. So let me take one of the focus of my work, which is actually the smoking-related diseases. So in Malaysia, I think in worldwide, this is one of the big issue. It was good that Malaysia have implemented the smoking ban at all food outlets, restaurant started on the 1st January of 2019. And Malaysia also signed like kind of the treaty that by 2045, that we should be a smoking-free country. But that is still too long to go. So currently we are working on many issues, particularly these respiratory diseases due to the smoking related. For example, the most common lung and oral cancer. And other than that, of course, like COPD. And we know as well that based on many data that, for example, since the 1930s, the incidence of lung cancer has been rising due to the widespread use of cigarette smoking. And lung cancer is among the most prevalent cancers in the world and responsible for approximately 18% cancer death in 2018. And this is most lethal. And worse, it only being found what you call the as a late detection. Yeah. So in Malaysia, the 5-year survival rate for lung cancer has been the lowest among all the cancers, with only about 11% of lung cancer patients survive after 5 years of diagnosis. In terms of personally, I also have my uncle pass away due to lung cancer detected only less than 2 months, and even my neighbor last year, only 2 months detected of lung cancer and they passed away. So I do believe that this is actually one of the things that we always have to remember, especially to those old dads. Yeah, remember that your kids always love you. So stop smoking if you do. All right. So let me. Yeah. Okay. Now we are shifting to the pulmonary drug delivery, because previously we worked more on the topical application, like ibuprofen, because we focus more on the and also the parenteral. But I think when we talk about the conventional drug delivery, I think many people also have spoke about this increased side effect, the hepatic metabolism, and there are so many other things, particularly affecting normal cell, where we can see that most of the patient who underwent the chemotherapy having a lots of side effect. And another issue that related to this delivery of anti-cancer, that most of the drugs are hydrophobic, very low solubility, and end up we need to use a very high percentage of or high dosage. Yeah, but the best thing about the pulmonary drug delivery, and if we're talking about the aerosol, yeah, particularly, we have to look at the best that what God created to us, which is actually the lung, have a large surface area to surface to volume area, and high permeability as well. Yeah, and good blood supply. I think this is one of the advantage why we should be using aerosolized pulmonary delivery. Yeah. So from there, for example, we can the advantage like even and well distributed. And I do believe when we work on this at this point, we've been approached that our formulation can also be used for COVID-19, but not in particular. We are talking about the delivery of the drug itself, but I will show to you later that we also use curcumin that may be what you call this to heal the inflammatory of the COVID-19 patient. Yeah. So among we are not working on the new drugs, even though a few of my group member working on the several new products, natural products particularly. But in our case, we really like to repurpose existing of anti-cancer drug, particularly like docetaxel, gemcitabine, cisplatin, afatinib. Yeah, this one is actually one of the latest one. But some, particularly docetaxel, that being used in most of what you call this treatment in Malaysia. In, for example, they can also use a dual drug, docetaxel and gemcitabine, docetaxel, cisplatin, or dual drug gem and cisplatin. But we also like to introduce the usage of quercetin and curcumin in our formulation. Yeah, we know that what the taxane and flavonoids normally being used, but of course they have a very poor solubility, yeah, and low stability, and worse, if taken in a higher dosage, it may end up with the development of drug resistance towards the cancerous cell, which significantly reduce the efficacy of the chemotherapeutic based treatment. So for this, what we work is actually on the lipid-based nanoparticle and inorganic nanoparticles, because my colleague from other university, from our consortium, working more on the polymeric nanoparticles. They're working on certain polymer. I cannot remember the name of the polymer right now, and they're also working on the chitosan. So we are working on slightly different. So I hope that I can get some of those work that we have carried out presented today, if I have the time. So the first part will be on the nano emulsion. We're working on the oil in water and also water in oil for single drug. For dual drugs, niazome, and we also use the iron oxide with the fish oil, but the focus is more on the solid lipid emulsion. Then we're also working on the powder, iron oxide, siRNA, iron oxide and reduced graphene, and then the MSN and MOF. Yeah, in particular, we also have these that related to dianomagnetosol. But however, because due to the COVID-19, there are many works that cannot be completed, particularly at the MRI test. So I'm not going to deliberate most of our iron oxide nanoparticle. And I think from the discussion for the past 2 days, I think everybody's talking about very much related to this. Yeah. So let me start with the very basic way of what we are doing. First, of course, we need to understand the screening of oil and surfactant. We did so many. This is part of it. Yeah, we screen many of the oils, including our own esters. Yeah, this is actually the difference between most of the work that have been published before. Normally people use oil, either soybean oil or the many different type of oil. So we try to use ester, and it works very well to our system. Of course, the surfactant, we need to find a good surfactant so that it will not increase the toxicity, even though that we want to solubilize as much as a drug as we can, but there is issue with the toxicity. So when we started from scratch, yeah, there is another thing that we need to be considered, the particle size. We then able to get below than 200 nanometer, particularly in between 100 to 150 nanometer. But another challenge is to get the VMD, yeah, volume mean diameter, which is actually the particle size. I will explain this shortly. So from here, we did a lots of things as well that we optimize it. And from the optimization, as you can see that the nano emulsion droplets that we managed to get basically a spherical shape, and the size obtained in between 100 nanometer, as you can see from this TEM micrograph. And it's also correlated with our measurement from the zeta sizer. Other than that, we must always look at the pH, yeah, because it is very important, not just about the delivery in what you call this in the tumor affected area, which is more acidic. But if it is too acidic or too basic, we're having problem when the patient have to cough, yeah, because of the irritation throughout the esophagus. Osmolality have to be one of our major concern as well, so that it is suitable and also it's stable throughout the system. And of course, the zeta potential also have to be, we also still focus on this because we do not want our formulation to be aggregated. We have to make sure it is stable at least for 90 days. So there are works that we carry out up to 6 months, but normally for the standard, we run it up to 90 days or 3 months. This is another thing. Of course, when we do this, we need to do it fast. We need to get more information. So other than MED, we also use artificial neural network, because from there we've been able to reduce the size of the VMD to around to below than 5 micrometer. Yeah. Then we start to look at the aerosol dispersion. There are many techniques that you can use to look at the aerosol dispersion. We use a spray tag. Spray tag. So the difference we can see from here that the percent disperse and percent inhale is not much different, and our optimum tree or the formulation tree basically give the highest, yeah, reach more than 80%. So it is consistent, and so from here we can do more testing. And if you look at another what you call this is the MMAD, yeah, the mass median aerodynamic diameter right now is already 3.09 for this formulation 3, which is actually good. And for the fine particle fraction, it can reach more to 90%. So this is very important, so that it can have a very optimal deposition into the lower airways, or we can basically talk about alveolar. Yeah. So this is just some other information. Now we can see that using the cascade impactor analysis, we can see the formulation can go basically to the stage 4 and 5, and some also deposited in stage 6. If you look at it, it's actually around here and up to the down to here, alveolar and alveoli. It is very important as well. If it is more than 5 or even maybe 10, it will deposit in the initial impaction, which is actually more towards this area. Yeah, but if we are working with the oral cancer, I think it is good as well to attack, or basically the drug to be deposited around this area. Yeah, because we have seen that normally people that having the lung cancer, they have a higher chance to get the oral cancer, or vice versa. But if it's too small, then it is too small to be exhaled, and normally it will sputter out, yeah, automatically. So that again, it is not good, because it will create the toxicity around the whoever that working in that area. So this is one of the difficult problem that we face. But in comparison with just quercetin alone, which is actually less than 10%, we've been able to release the drug at the almost 25% at the different for this formulation 3, and it basically fitted the what you call these Cosamaias peppers, and it showed that it have the sustained release behavior, particularly in acidic environment. Yeah, so it increased the rate of destabilization and then disrupt the liquid film between the nano emulsion droplets. And based on the IC50 data, we can see that at 48 hours, it was about 300 microgram per ml, and followed by a reduction to 100 at 72 hours. And again, this can be attributed to the sustained release of quercetin from nanoemulsion, and probably due to the internalization and increased availability of quercetin for a longer period, in which again, it can be correlated with the in vitro release study, that I showed here. Okay, now I use another nanoemulsion, which is actually water in oil, gemcitabine. The process is very similar, except this one, what we can say that is a totally different way or different approach. The idea is to get as much as gemcitabine into the system. So by using the optimal mixture design, we can see that we maintain, or we manage to get below than 150 nanometer. And then the consistent with the pH value and also the zeta potential. So this again showed that no separation after 3 months and even at the 3 different temperatures, which is actually quite consistent. So when we talk about the drug release, the drug release is actually almost threefold at the pH 6.5. And again, this and if we relate this to the cytotoxicity study, it showed that the optimized nanoemulsion containing gemcitabine induced the cytotoxicity toward the A549 lung cancer cell, and at the same time reduce the cytotoxicity towards the MRC5, which is actually the healthy cell. Okay, so I would like to bring you to the another nanoemulsion that we formulated. It contains the docetaxel and curcumin, and we know that both are very hydrophobic. And curcumin, I think in Asia we are very familiar with the curcumin. Was also identified in our target. Our work, it is more as a chemosensitizer rather than as anti-cancer itself, whereby the lung cancer can be sensitized towards this chemotherapy. So we believe that our result also showed that the synergistic anti-cancer effect exhibited by docetaxel and curcumin may potentiate the treatment of lung cancer and become an attractive therapeutic drug candidate for both. So same as well, we are looking at the influence of the composition. I think this is very important to look at this, but in our case, we are very focused into the aerodynamic size, where we observe these diligently. And other than that, we're also looking at the process parameter. In this work, we use a sonicator, so in order to get the consistent size. And what I will show you later on, we're able to study as well the different sizes of our nanoemulsion at 100, 150, and 200 nanometer, because we heard so many claims that the lower the size, it may give you a different toxicity because of the nano environment. So we try to prove whether it is related to the claim or not. So from here, we studied 3 different sizes for each emulsion. Either it is only docetaxel alone, curcumin alone, and mixture of docetaxel and curcumin. There are too many data that I don't think so I'm able to elaborate each, but what I can say from here, because we are looking at the aerodynamical properties, the pH, the osmolality is very important, because it may increase the drug loss if we not able to control it. Result in the bronchoconstriction, irritation, and severe cough. I think this is part of that I have explained previously. Just like to show that we will still be able to focus on the aerodynamical properties so that we can use for our work later on. So this is the cascade impactor analysis, and this is particularly for each different sizes. And as you can see that, excuse me. Okay, before I elaborate more into detail, our our limitation is actually on the nebulizer itself. If we can get a better nebulizer, we believe that we can get a different type of sizes. Yeah, and from here we can see that for the 100, most of what you call this the nano emulsion deposited in stage 3, 4, 5, and is quite high as well in the stage 6. And for the combination, same as well. And we can see that the size play a very important role, that the 100 is actually the highest, can penetrate into the, can enter into the stage 3, 4, and 5, yeah, the deposited and that particular area. So from here, I'm just going to go quick into the study of the drug release based on these. Same as well, that we found the acidic environment is very much related, because the 6.5 is better than 7.4. But other than that, the drug release profiles of the encapsulated drugs were found to be relatively faster compared to the free drug solubilized. In our case, we use the simulated lung fluid, yeah, at the pH 6.5. So the result of the optimized nano emulsion with appropriate physical and aerodynamic properties for inhalation also showed that is a sustained drug release manner might suggest that this can be a good or potential application. And of course, the effect of size 100 nanometer can be seen very clearly in this work. So we look at now on the in vitro cytotoxicity. This is the MTT test. Again, we can see that in combination of DTX and curcumin, able to inhibit cancer cell proliferation better than single drug, as in the IC50 is actually the lowest. So again, docetaxel and curcumin both can be synergistically inhibited the proliferation of A509, in which CCM play a major role as a chemosensitizer. So we also look at the ex vivo nanotoxicity, and this is in collaboration with Harvard. So my postdoc work this in Harvard because this is a consortium of Malaysian project with Harvard. So we are working with the Professor Brain. So when I visited them, so they're working on the precision cut lung slice, looking at nanotoxicity. We did the independent research in Malaysia and also in Harvard. And from his work, we again confirm that the size of 100 nanometer basically give the most significant or increase significantly, yeah, in high concentration. But however, the free drug solution and drug-loaded nano-emulsion with the size of 100 and 200 nanometer did not affect that much when we talk about the tissue viability. So this is again interesting information, and from the information we dig into details what's going on. So using a zebrafish, we studied the permeability of the different sizes, and from here we can also see that the 100 nanometer size able to permeate through the chorion as early as 1 HPF, yeah, and exhibited highest fluorescent intensity compared to the other formulation. Yeah, can also see from here that this is actually at the outer membrane. So fluorescence was observed at the embryo's outer membrane in the aqueous solution. So again, we like to highlight that the tissue viability of 100 is lower than 150 and 200, and also the control. This might be due to high drug concentration able to permeate in the lung tissue as supported in the permeability study. So we then continued to look at the subacute nanotoxicity using a zebrafish. So the presence of heterogeneity in the developing larvae significantly increased with increasing concentration of the drugs, and varies among the different sizes as well of the nano emulsion, where we can see that several morphological defects such as short body length, curved tail, bent tail tip, and many more from from the observation, I think in detail, because we have to look into one by one. So therefore, we can see that the scoring for the acute toxicity assessed with regard to the survival and morphological changes is actually as depicted here. Yep. Which is we are looking at the LC, the lethal concentration value for each nanoemulsion samples. And again, we can prove that the size of 100 nanometer giving the lowest value compared to the other size. So beside that, the different sizes of nanoemulsion also have demonstrated to increase the LC50 compared to the single drug. So these results were in line with our previous in vitro and ex vivo nanotoxicity studies. Okay, so we also did other works on dual drug, gemcitabine and cisplatin, the most common that are being used in Malaysia for the treatment. But in this case, we work for the niosome. It's a very similar approach that we carry out. Just like to highlight that based on this work, we're able to get the correct size. And from here also, we have shown, as we published recently, January this year, that the result also showed that the optimized niosome gemcitabine cisplatin had reduced the cytotoxicity effect against both MRC5 and A549, but in this case, of course, is better for the A549. I did mention that we also run for the iron oxide, but I'm not going to explain that one in detail, except one of these, because this is slightly different. Because another one just now is a quantum dot, so very similar to the speakers previously. So in this case, we use a super paramagnetic graphene-based magnetic nano composite, which is actually synthesized via in situ chemical approach. And this nano composite later on be used as the drug carrier, so that we hope it can be guided by external magnetic field to the specific site of the interest for targeted drug delivery application. We actually want to use this for the breast cancer, because for the lung cancer, it may cause a lots of problem because of when you want to guide with the MRI. So it is actually quite difficult when the alveoli is actually quite deep into the body. So we can try this for the breast cancer. So in this work, we use Ganoderma lucidum extract, which is actually able to stabilize the RGO, the reduced graphene oxide, iron oxide via the hydrogen bonding. And one of the big issue when we work with the graphene is actually high toxicity. So we also use pluronic F127 to reduce the overall toxicity. Other than that, the best thing when you use the polysaccharide, the hydroxyl function group on this polysaccharide exhibit the hydrophilic property, and then it may able to the dispersibility and also the stabilization was enhanced tremendously. If you can see from here that after 15 minutes, that for the dispersibility is still very good, but without the GL, you can see the deposition or sedimentation of the iron oxide after 15 minutes. So this finding, when we talk about, we also completed this study, we can also see that the complete system displayed high toxicity on A549 at IC50 of 13.9 and almost 10-fold lower compared to the MRC5. So this finding can be explained based on the fact that polysaccharide is also exhibiting anti-cancer properties, which is capable to inhibit the growth of cancer cells. For this MSN, I'm not going to deliberate more on the activity, but more into the even though we use this for the what you call these drug loading and drug release, but I'm just going to highlight that this is one of the very high surface area MSN where we use ionic liquids as a template, And from there, we study the effect of alkyl change length and also the anions. Yeah, and we use quercetin for this work, and currently still continue in collaboration with this one is headed by my mentee Haslina, working with the Oxford group. This is what we've been tasked to do for the past 3 years as we work closely with Professor Omayagi. He attended UPM, there is a Mustafa Award, Nano Materials, way back in 2016. And after that, the UPM honored him as the honorary doctorate, and from there, that's why I was in Berkeley to work on the metal organic framework. It's very new to us, but Alhamdulillah, we did quite a good job. We managed to get many things work in a very short time, but it's very, very, very difficult for us because the challenge is to design and to synthesize our own morph or ZIF. So I do believe that maybe some of you already know about morph of ZIF, but in general, it's beautiful because when you use a metal and organic linker, there is no limitation. Yeah, you can do anything, and even if it is known with a different combination, you might get a new or different topology or crystal structure. So I would like to share with you our latest work, which is just published in Dalton Transaction, where we establish a versatile nanoparticle through a straightforward method. We change the common method, which is actually high temperature. So in situ, including the gemcitabine, and in this case, we conjugated with the RGD. Yeah, in this case, the RGD is actually the autonomous homing system of this peptide, arginine, glycine, and aspartic, is via a one-pot solver thermal reaction. So in this work, we can show that the RGD peptide is a binding ligand that homes the integrin receptor alpha B beta 3, which are overexpressed on tumoral endothelia. Yeah, in that case, the RGD functionalized and ZIF or nano ZIF, we're able to get a very small ZIF below the 100 nanometer, carrying a chemotherapeutic payload, in this case gemcitabine. Then the reticular nanoparticle then can dock on the surface of the cell to deliver the agents that are being carried out. So once it reaches the tumoral endothelium surface, the cellular uptake will occur, and then the gemcitabine induce the cytotoxicity that leads to the cell death and ultimately cellular apoptosis. So we have shown that the successful functionalization of the surface with RGD was proven by spectroscopic and electronic microscopy techniques, but I do not show you all the XRD, FTIR, and so on. Yeah. So in this case, what we like to show to you is that in order to determine what you call this is how it looks like. Yeah, the untreated, and then when we put the fluorescein into the system, because we want to see what's going on to determine the cellular uptake efficiency, we replace the gemcitabine with the fluorescent dye fluorescein, and then from there we monitor based on this. We can see that the highest fluorescent intensity after 2 hours actually in this. Yeah, you can see that aggregates and accumulate on the cell surface, which indicate endothelial cell attachment, greatly reducing the exotisis process. And from here as well, the data also show that based on this, that the system that we introduced with the gemcitabine, experience not only efficient uptake within the A519, but also induce obvious cytotoxicity, about 75% at a concentration of 10 microgram. And then apoptosis about 62% after 48 hours treatment when compared to the nanoparticle. And we are so great as well that this paper has been selected as the cover page for the recent Dalton transaction. And for me, it is like kind of a beautiful gift to give to my university, who also celebrating the 50 years of UPM and Dalton transaction also the 50 years anniversary volume. And currently we are working with other different MOF like 74, MOF 101 and ZIF 90. The beauty about the ZIF 90 because it have more places for the conjugation to take place. And we also working with our own MOF, the UPM MOF 1 and 2, and the difference between all, we are working on the S block metal, which is actually more biocompatible compared to the MOF or ZIF that have been used in the most publication. So currently we also looking at the not many people working on the AFM, yeah, for MOF. So we thought that why don't, since we have this equipment, so we thought that why don't we look at this and we can show that the permeability and nanotoxicity can be very much related to the roughness of the nanomaterial that we can see, yeah, for this one is actually the highest, it being able to penetrate the cell membrane. And based on our zebrafish, embryo zebrafish work, we can also again see that the permeability assessment is actually the highest for the RGD peptides, nanoparticle. So in conclusion, we can say that our ester-based nanoemulsion can be a good nanocarrier either for single or dual hydrophobic drugs for pulmonary delivery system. That also we can talk about the aerosol. Yeah, suggested aerosol formation suggested that potential pulmonary route, yeah, to reach even to the alveolar region of the lung. And the most important is how to control the nanoparticle size, yeah, because it may affect the nanotoxicity of the cell. It is actually interesting to study this one in detail. And we also hope that to work with a new material as nano hubs for potential drug nano delivery. So before I pass it back to the Mr. Chairman, I would like to acknowledge many of my team members. Many of them are my mentees. Dr. Azlina Liza, I think she's here with us to right now, and Dr. Azura she's working on the nano emulsion, yeah. And then we also like to thank Prof. Joseph Brain. He's really into the respiratory, and of course Professor Omayagi working on MOF, and Carl Cordova. He was from Berkeley, but now is in the Royal Scientific Society, Jordan. Here are some of my students. Most of the work that I presented just now. I think Akma is here. She's waiting for her viva. And I mentioned earlier that this is our work is actually sponsored by the Nanomite or Malaysia Institute for Innovative Nanotechnology under the Ministry of Higher Education. Okay, so I think I would like to thank everybody. Might be slightly different. Somehow in Malaysia we have more women, yeah, in our science lab compared to the male. It's very difficult to get all these species right now. So we always welcome for collaboration between Malaysia and Pakistan, inshallah. Thank you very much for listening to basically more on the sharing of what we done in Malaysia. Thank you. Assalamualaikum.
Very much, Professor Abdurrahman. Very informative lecture. Really enjoyed. So we have time for the questions now from the audience, please. Over there.
Thank you very much, Professor Rahman, for your nice and innovative talk. I have just a quick question regarding the surfactant in the lungs and their compatibility or their action with the nanoparticles. Can you comment on that?
Okay, yeah, I think that's a very good question, and this is what we actually wanted to do. At the moment, we are waiting for we actually want to do the in vivo, but we've been halted by the, what you call this, the pandemic. So it's not like I don't know what to answer that question, but because we do not see yet what happened actually, so I think I might like to reserve what you call this, the answer, because we found that it may be different between one nanoparticle or even one or nano emulsion, how it affect into the lung system. Yeah, it is actually quite tricky. We are actually hoping to work on small and big animals because we have the veterinary medicine faculty. So because there are many dogs have been affected with the lung cancer. So yeah, that's what I can say at the moment. I'm not able to deliberate in detail because I'm afraid that it may give a false answer because we haven't tested it yet and come to the detail in vivo.
Thank you. So any other questions? One is back there, please.
Assalamu alaikum.
Waalaikumsalam.
Dr. Abdul Rahman, it is very great presentation. However, I have a question. Actually, I'm not familiar with the term aerodynamic particle size that you have mentioned. But we have, I am more familiar with hydrodynamic particle size. So is there any difference between aerodynamic particle size and hydrodynamic particle size?
Yeah, aerodynamic is actually, if I can show it here, yeah, aerodynamic is basically we are working on the basically a droplets. When, for example, if you cough, then you can see the particles. That's why I think for the COVID-19, people have to stand at least 1 meter or better 2 meters away, because that is actually the aerodynamic, what you call this, aerodynamic properties that we are looking at. So if it is too big, normally it's heavy, so it will deposit it slightly closer. When we talk about the what you call this, the inhalation, there are many techniques of inhalation. Normally for the lung cancer patient, they do not have the strength to inhale, yeah, because of the maybe the lung is very constrict. So there are many ways. Either you use a nebulizer. So that's why I mentioned just now, our limitation is actually the nebulizer. Okay, so when we talk about the aerodynamic, the properties, that the consideration is actually the size. Either is small or it is big. But when we talk about if it is too small, yeah, for example, less than 1 micron, so basically it can float, or even if you what you call, you just imagine that if normally kids with the asthma, they have to take the inhaler. Yeah, if it is too small, it will exhale back. So that's why we are particularly looking at the aerodynamic size. I'll share with you the what I meant by that. Yeah, okay. So we hope to get the higher concentration, the higher size in this particular, because we want to reduce the too small aerodynamic size. Yeah, aerodynamical properties, and how fast as well it can explode, it can go. Because when you press the nebulizer or the inhaler, this method we use a spray tag by Melvin. So how fast and also how far that the particle can come up from the nozzle. Yeah. So, and based on that, that's why we look at other properties here. For example, this FPF, which is actually the fraction of particle, how much that is actually can be released. Some of the nebulizer may not be able to give a full release. Yeah. For example, in this case, we're able to get about 80 to 84%. But in our previous case, we're able to get to 90%. And many cases, not able to reach even 70%, meaning that there are still many drugs left in the nano emulsion or in the solution. So basically, we are taking more all of the stabilizer rather than the drug itself. So I think this is what we're talking about. The VMD is actually the size of the particle or the aerodynamic. Yeah.
Thank you very much for the detailed answer, Professor
Okay.
Ahmed. Another question.
You're welcome.
please.
Okay, so thank you, Professor Basiruddin, for a very informative talk. So I want to ask you that as we see that the smaller size nanoemulsions, they increase the drug bioavailability, but at the same time, it increases the toxicity, as showed in your zebra fish results. So is that possible that we can just make a replacement of the nanoemulsions by N polymer nanoparticles, so we can just solve this issue and decrease the toxicity? Because it is organic in nature. So any comments?
Yeah, this is what we are looking at currently. Our what you call this, our formulation right now being taken by our Institute of Medical Research to look at in detail, and they also suggested that if we can encapsulate to make it slightly bigger, because we know that the smaller the particles, then it may fast the microphage, yeah, but normally is too low, maybe below than 10 or 20 nanometer. So we are quite surprised to look at this, and Harvard is taking charge to understand more when they want to do the detail of this in vivo. But as you said, yes, we are also opting to use other what you call this in order to reduce the toxicity, even though the penetration is good, but the toxicity is too high. Yeah, as you suggested, we are working on that. But at the moment, we're also working with another two. Yeah.
Yeah, thank you very much. Very, very interesting questions and answer session. Okay, is it a quick one? We have last very burning question.
Assalamu alaikum. I'm Dr. Naveed from Qaeda. Professor Rahman, very nice talk, and I think it's you selected a very tricky system, that is the delivery of the drugs through the respiratory system. It's very difficult to deliver drugs through the system. The things which are, how to say that the questions are being raised because still there is the in vivo study is pending. And I think you mentioned that to avoid the exhalation of the smaller particles, you will go for the larger size particles. I think there is another mechanism of the respiratory system which can be difficult for the deposition of the large size particles. That is the mucociliary clearance. And when you increase the size of your particles, the mucociliary clearance will not allow you to deposit your particles in the respiratory system. And kindly think about this when you are going to the results of the studies of the in vivo analysis. Thank you. Thank you.
Yeah, thank you. Thank you very much for the suggestion. Actually, we met to be honest, I'm still a chemist. I'm not a pharmacist or with a biomedical background. But when we sit with the medical team, including the expert, this is what they highlighted. In fact, the issue about the mucus and what you mentioned just now is actually correct. And that's why we want to study this in detail using the correct animal study, and by inhalation. We have the bottleneck when come to the because not basically we are maybe the first one to carry out this work in Malaysia. So we have difficulty to find such equipment, and those I forget to mention that for all of our powder is not yet been tested for the aerosol delivery. Yeah, so that's again another limitation that we are working on. As I said just now, we started this work for the what you call this for the nanoparticles or the solid part for the past 3 years. So there are so many things that we need to learn, and inshallah, with all the comments just now, we take into consideration, and in fact we want to do it. Yeah, and we also welcome any collaboration.
Rahman, for joining us from the Malaysia. Really appreciate your time, and it's a really good, very critical question answer session, and you responded very well. So ladies and gentlemen, join me to thank Professor Abdur Rahman.
Bismillahirrahmanirrahim. It's my pleasure to introduce Dr. Yar, who was my student when he was quite young, but he is still quite young. He obtained his PhD degree from Bristol, UK, and postdoc from Nagoya University, Japan. He has published already 76 articles in leading international journals. He is having 14 international patents to his credit. And he is basically working at ComSets Lahore campus. He is associate professor at Biomaterial and Tissue Engineering. He is also head of the Skin Tissue Engineering group. His work is more like applied oriented, and he has collaboration with several other groups from across the world, like in UK, MIT, and other part. So he would be talking about a history of our working on pro-angiogenic agents, developing interdisciplinary research, and that can translate to the clinic. So, Dr. Yar, it's over to you.
Thank you very much, organizers, especially Professor Dr. Iqbal Chaudhry and Professor Dr. Irshad Hussain and Professor Raza Shah, my teacher. Thanks for the kind introduction, and is really pleased to join this very informative workshop since yesterday. Since we have been learning a lot about the nanotechnology, and we are learning that how we can apply this nanotechnology to solve the issues which we are facing at the moment. So what I will be talking about over next 30 minutes time is about the journey, that what was the challenge we took, and how we were able to deliver this, and what stage we are at. So basically, I will be talking about history of our working on pro-angiogenic agents which stimulate new blood vessels formations, and this new blood vessels formation is really critical when the wounds are not healing. And this sort of interdisciplinary research, because we are working with clinicians, industries, and a lot of other labs, and that how we can translate this to the clinic. Very challenging working in Pakistan, everybody knows, but it's not impossible. So working with the team with the University of Sheffield and the other partners I will be talking about. So while we are working on the wounds, there is a good business opportunities developing advanced wound care management products. For example, if we look at this digit, 19.8 billion business back in 2019, which is expected to reach 24.8 billion by 2014. So this is clear that this is big business opportunity. So why we are interested in this area, surgeons treating patients with burns do not have access to the advanced wound care products or tissue engineering therapies, and patients with chronic wounds, they are facing a lot of problems because they are not and amputations going on. High technology solutions such as tissue engineering and regenerative medicines at the moment is not feasible in Pakistan due to very high cost and non-availability of infrastructure in Pakistan. However, development of the dressings or materials which can stimulate the can solve a lot of problems. So this is the contents of my presentation, our history of working on angiogenic agents. This is tiny sugar molecule, and I will be talking about this, that how good this molecule is actually, and does it 2-deoxyderibose compared to the VEGF. VEGF is the protein responsible for angiogenesis in our body. This is present inside the body, but this is not sufficient when the wounds are chronic or the wound size is very big. How does it work? How much we understand R&D from the industries, our plans to move this product to the clinical benefit, basically. This is very important slide. This actually shows that how we design such products. So very, very difficult to gather such people. So this is Professor Rashid Bashir. He's heading Mayo Hospital Burn Unit. Professor Amr, he's heading burn unit at Civil Hospital Karachi. Professor Mughis, he's the leading guy when we heard about Ahmedpur Sharkia incident. He was the professor accepting all these patients in his unit. Professor Tariq Iqbal, he's heading burn unit at PIMS Hospital, Islamabad. So we were able to, luckily, to gather all these very busy clinicians under the roof, and he's the industry guy. So what was the question we asked them? What you people need really urgent in your burn centers? And what you want us to develop. So the whole story comes from here, that what they actually needed. So basically understanding the patient needs, that's the previous slides tells, that we want and go to the clinicians and ask them what you need. And then we design and produce the biomaterials, testing of materials in relevant animal models, and obtaining regulatory approval. That's a very, very critical stage. And we were happy to see Dr. Abdurrashid yesterday here from the regulators. It's very important to work with the regulators and tell them what we are doing and what they think about this. And then next is the first in man safety study. And after this, larger clinical trials, phase 3, phase 4, and then there is the industrial production. So vascularization, that is the new blood vessels formation, is required for wound healing and for the translation of tissue engineering construct to the clinic. Their source of oxygen and nutrients to the wound bed, and vascular endothelial growth factor is responsible for the angiogenesis naturally. So this slide shows that how we are started from utilizing nanoparticles. In 2015, the paper was published on zinc oxide nanoparticles. Being organic chemist, I thought that zinc oxide basically produces hydrogen peroxide on the spot that stimulates production of the VEGF, and that zinc oxide was supporting new blood vessels formation. Then we have prepared zinc peroxide in collaboration with Dr. Nasser at our center. My student Samreen, she was working on this. And then we tested this in the CAM assay. I will be talking about in a while that what this CAM assay is in the detail. And we were able to see that zinc peroxide was better as compared to zinc oxide in supporting new blood vessels formation. This is paper. We were collaborating with Qatar University. What we have done is that that if we can supply oxygen to the wound site, this oxygen can be really helpful in the hypoxia conditions where there is no oxygen was available. So what we have done is we have took PCl and then loaded with sodium percarbonate and electrospun this. And in vitro, we have tested this was releasing oxygen. And then we have tested this in collaboration with the Center of Excellence in Molecular Biology at Punjab University. So they have tested this at protein and gene level, that this was stimulating the production of HIF, that is the factor responsible for enhancing enhancing the wound healing. We have tested in CAM assay and then tested in this full thickness wound model as well. That what we have seen is in the case where these dressings were loaded with sodium percarbonate, there's a proper formation of the dermis and epidermis, and we can say hair follicles as well, and larger blood vessels as well. In 2014, luckily, a team from University of Sheffield visited us, and they selected five fellows for Pakistan UK fellowship sponsored by ComSearch University, and luckily I was selected as well. So I visited the University of Sheffield, and from where this story started. So we have developed we have found that this 2-deoxy-D-ribose, basically this is part of DNA. When the DNA breaks down, we end up with 2-deoxy-D-ribose phosphate, and this 2-deoxy-D-ribose phosphate starts a cascade of the reaction, and that end up with the stimulation of the angiogenesis. So I just thought, if the body is doing like this, if we supply this externally, whether this can support angiogenesis. So very quickly, I made this electrospun during my Sheffield visit, and we can load this into a variety of dressings. Basically, this can come into hydrogels, gels, electrospun membranes, and many more carriers as well. So we made these porous materials, tested in CAM assay, that we have found this was supporting new blood vessel formation, tested in animal trials as well. That there's a very interesting results. This was open wound still at day 17, and this was controlled, just such membranes without 2-deoxy-D-ribose. When this contained 2-deoxy-D-ribose, this was properly closed. We have done a lot of stainings. These coming slides will be about this 2-deoxy-D-ribose, that why we need 2-deoxy-D-ribose, why not VGF? If we look at the price, 2-deoxy-ribose effective solution will cost very small amount of money. This is 0.08 pounds. But if we look at the VGF, this is more 2,000 pounds, very expensive. Stability, everybody knows the proteins and growth factors and biological stuff is unstable. And if we talk about this 2-deoxy-D-ribose, this is stable for at least for 2 to 7 days at 2 to 8 degrees centigrade as a solution. And there was companies, they have started producing the product for difficult to heal wounds by using VGF. But there was a lot of problems supplying VGF externally. They have seen this leaky blood vessels and some other related problems of cost and stability as well. So from now on, I will be talking about our in vitro results. We use this human aortic endothelial cells. Sarkan here, PhD student at the University of Sheffield. So on the day 1, we have found not significant difference. Everything was good. On day 4, that one of the concentration was not good. It's 110 millimolar concentration of 2-deoxy-D-ribose. But all the concentrations were good. On day 7, what we have seen is that one of these concentration, 100 micromolar, that was equally good as VGF. So these are really interesting results. So we have compared this metabolic activity with the number of cells, and we have found this consistent, that 100 micromolar concentration was equally good like VGF. And in here, we have compared with the other sugars that how other sugars will be doing. So what we have found is that on day 4, that this 2-deoxy-D-ribose 100 micromolar concentration was equally good like VGF. And on day 7, again, this was good, 2-deoxy-D-ribose. But the other sugars like 2-deoxy-L-ribose and D-glucose, they were not effective. We have seen not significant difference between the control and these sugars. At this stage, we're interested in to learn that how this tiny sugar molecule is supporting blood vessels formation. So we took the cells, and these cells were treated with 2-deoxy-D-ribose, 100 micromolar and 1 millimolar, and also these sugars when released from variety of the dressings. So what we have seen is that these sugars resulted in the increase of VGF. And we know that VGF is responsible for new blood vessels formation. So from here, we got this confirmation that this 2-deoxy-D-ribose is supporting new blood vessels formation by upregulation of VGF. So the other thing was that how this sugar will be metabolized by the cells. So we took the cells and treated with 2-deoxy-D-ribose, that we have seen that this sugar concentration was decreased over the time in the media, and this reduction was more when the cells were present. It means cells were eating this 2-deoxy-D-ribose, which was good that over the time this will be metabolized by the body. So from now on, these slides will show that what is this Cori-Allen-type membrane assay, very useful assay. This is chicken fertilized egg. This is the stage at day 3, and look at day 7. There is aggressive formation of new blood formation. So if we want to study our material, whether supporting new blood vessels formation or not, just imagine if we put our material here, and this material, if it is itself angiogenic, or if it is releasing something which is angiogenic, so we will be able to see a lot more blood vessels formation here. Or if it is anti-angiogenic, this will stop the blood vessel formation in this area. Very useful assay. We are using this. And this is how our samples look like at day 14 when the experiment is finished. This is very interesting slides that this egg was treated with PBS control, and this was treated with 2-deoxy-D-ribose, 200 micromolar per day per embryo. This was the stage we were trying to understand that how much dose of 2-deoxy-D we will need to load on the dressings. So what we have seen is this concentration increased around 3.6-fold in the angiogenesis as compared to the PBS. And we were really interested in to see whether 2-deoxyderibose is resulting proper blood vessels, not leaky and porous as well. So what we have seen is that we have injected this rhodamine labeled dye into the circulatory system, and we have seen these proper blood vessels, porous and interconnected as well by using 2-deoxyderibose. So at this stage, we have also produced electrospun fibers, and variable concentration of 2-deoxyderibose was loaded. This is the case where the control, there was no 2-deoxyderibose, and the size was 0.66 micron. And when 250 milligrams, the size was increased, and this was further increased with 500 milligram of 2-deoxyderibose. Basically, we were interested in to see which concentration is good. So look at this, that this is the number of blood, this is control anyway, there is no sugar. And this is the case where it's 250 milligrams of the sugars. So we can a lot more blood vessels here. 500 milligram was not good. So based on this, based on this data, we have proved that this lower concentration of 2-deoxyderibose was better. So this was the stage when one of the Pakistani company, they were really interested in to upgrade their dressings. Two of the dressings, we know that this cotton gauze, this is available everywhere. Whenever there is an injury, we will end up with cotton gauze, the first line of the treatment in Pakistan. They were interested in to upgrade their two products. One was the wax-coated cotton dressing. This is called the Paratule. This is being used in the burn centers very frequently. This is not. The other one is non-woven fabrics, and this is better as compared to the simple gauze because this is non-woven, so it's relatively less stick to the wounds. So we took these materials from the industry, and they sponsored us this research basically. We asked them just sponsor us this money. At the end of the day, if you're really interested, so you have rights to take this technology onward to the market. So we have seen that 150 microgram per meter squared, relatively thick fabric was suitable, and we have seen that this was able to release the 2-deoxyderibose over 5 days of time. This is the Biles assay we use to detect that how much 2-deoxyderibose is being released by using spectrophotometer. This table shows because a lot of formulations, so all formulations were not good. Either we were seeing a quick release of the sugar within an hour, or we haven't seen any release. So ideally, we need a system which can give us the release from the dressings over a few days time, because we need to change the dressing, for example, every week or every 3 days or something. So what we have found was that just dilute wax mixture by increasing the liquid paraffin, we were able to see we get this release 5 days time, and within the right dose. What is the right dose? Or by using extensive cellular studies, chem assays, what we have found is that if we apply 250 microgram to 500 microgram is the effective concentration of this 2-deoxyderibose. So we were able to achieve this concentration from this formulation. We have tested this chem assay, extensive histological studies. They were supporting new blood vessels formations, and healing was good. We have published this with the industry. Industry was happy that one of their author was on this paper. Very rare example in Pakistan where we publishing together with the industry. About this, what is the future of this? I will be talking later that what stage this is. So back to the in vivo assessment. We were really interested in to see that whether this technology will be effective for the diabetic wounds. They're relatively difficult to heal when the patients are with diabetes. We took this alginate dressings. These are very, very standard dressing, which is being used worldwide for healing wounds. We took two concentrations, 5% and 10% weight percent of the sugar, and tested this on the rat. What we have found is that on day 20, the wound which was treated with alginate, there was relatively close, but this was completely close when treated with alginate 5% and 10%. We've done extensive histological studies, H&E and CD34 to study the new blood vessels formation. And we have seen that with CD34, that relatively more number of blood vessels than when these were loaded with 2-deoxy-D-ribose, and similar pattern on day 20 as well. For implant rejection and activation of a chronic inflammatory response, we use this CD80 basically, and there was not bad news from any of these materials on day 7 and day 20. And similarly, we stained these materials with CD163, which is for constructive remodeling response. And we have seen that dressing, which is alginate, and also 5 and 10%, they were helping in constructive remodeling as well. So in conclusion, that what we have learned from this is that 2-deoxy-D-ribose 100 micromolar to 1 millimole has been found to be 90% as effective as VEGF. Very interesting. And although the mechanism action of 2-deoxy-D-ribose is not very clear, but we have recently shown that 2-deoxy-D-ribose increases the production of VEGF, which is responsible for blood vessels formation. So what ChemAssay has shown us is that 2-deoxy-D-ribose 200 microgram per day per embryo has been found to be 80% as effective as VEGF in inducing angiogenesis in ChemAssay. 2-deoxy-D-ribose can be easily loaded into and released from tissue engineering construct to stimulate angiogenesis. Basically, we have tested so far from a variety of the materials. Incorporation of 5% and 10% 2-deoxy-D-ribose to the alginate and various other dressings have shown that wound closure 100%, but stimulated angiogenesis in the wound area as well. So these were our conclusions. So how is our history on this? This history has spanned from working in Pakistan, UK, USA with funding from Turkey, Kinenta, new startup company based on this technology, Cottoncraft, Pakistan, and a joint patent between IRCBM Pakistan and the University of Sheffield. How does this compare to VEGF in activity and potency? About 80 to 100% like VEGF. And how does it work? How much do we understand it? These are all the questions we are trying to address. But at the moment, we have evidence that this supports upregulation of VEGF. These are papers which we have published together with our collaborators from UK, from USA, Turkey. And there was a good news coverage based on this when we have first time published this. Forget everything, you know, sugar could be good for you after all. So it's not the table sugar. This is very specific 2-deoxy-D-ribose sugar. It's good for you. The University of Sheffield highlight this, physical organization as well, in Pakistan, Outlook as well, Express in Urdu as well. Khas sugar gare zakhmon ke liye maram ka kirdar ada kar diya. This is very good to see that everybody in Pakistan was able to read it. In India as well. So we were really pleased that India covered the story as well, and MIT technology reviews. So we have where we are. We have licensed this technology to Kinenta 2 years ago. We have an agreement with them, University of Sheffield and ComSets, and now this Kinenta is working at a startup company. That's recently they've hired very good names, Professor Fiona Wood, one of the leading burn surgeon from Australia, and other personnel as well. So this basically I took this from their data, that how they are planning basically. So development is at the University of Sheffield and ComSets, clinical trials from University of Western Australia, and they are planning a lot of agreements with the world leading manufacturers of the dressings. Okay, so back to the dressing, which we were working with with Cottoncraft here for Pakistan, because our first aim is to introduce the products in Pakistan for our burn patients and diabetic foot ulcer patients. So very luckily, in collaboration with Professor Raza Shah Saab, we applied for a funding to the HEC and British Council. There were 260 applications, and we were among the 6 who won this grant. So just got this award letter 2 days ago, so it's good news. So we have funding. So we have very competitive team for this. We have ComSets, our laboratory, University of Sheffield, their dental school and Croto Research Institute, Mayo Hospital Burn Unit of PIMS Hospital, this Bioequivalence and Clinical Research Center, HEJ, is with us, and Cottoncroft is industrial partner. So what we will be doing with this, we have already loaded this 2-deoxy-D-ribose on cotton dressings, and they are all ready to be perform third-party toxicity trials under the leadership of Professor Raza Shah, and then we look forward to first-in-man clinical trials in over a few years' time. This is last to acknowledge Higher Education Commission, Industries Cottoncroft, Cananta, University of Sheffield, and students here in Pakistan and students in Sheffield, Professor Sheila McNeil as well. And thank you very much for your time and listening.
Okay, so thank you, Dr. Yar, for again a very good presentation. So we have time for question. Yes.
Assalamualaikum. My name is Mehreen Tariq, graduated from Comstech University, Islamabad, and currently studying in Qaid-e-Azam University, Department of Biochemistry. Thank you, sir, for such a thorough and excellent presentation. I have some basic questions. The first one is that you told 2-deoxy-D-ribose is a pre-angiogenic agent. So are there any findings that 2-deoxy-D-ribose can make a tissue prone towards tumor formation, since it is known that tumor formation includes formation of new blood vessels? And the second question is that is 2-deoxy-D-ribose produced in our cells under normal physiological conditions or any stress conditions? Thank you.
Okay, so it's a wonderful question. This was the first question when we were talking to the industry that exactly came back to us that whether this will trigger this reaction which can lead to the tumor. So the answer is that we haven't got any concrete view at the moment, but what we understand is with this data, we are confident that if we are supplying this sugar at this very small concentration, which is really required for the angiogenesis, and after this is metabolized by the body and gone off, so maybe not will lead to the tumor cells. But with this money, which we have recently got, we will be studying this in further details.
Thank you very much, Dr. Yar, for a nice presentation. I have two questions. What other parameters besides the blood vessel formation could be a source of identification that this 2DR technology is working for the angiogenesis? That is the first part of my question. The second part is that recently various double dressed and even triple dressed sodium alginate dressings, they are reported in the literature, not only from abroad but from the Pakistan as well. So they found really good efficacy against the diabetic induced model and the Burns model. So this is my suggestion and a question as well, that whether you apply double or triple dressing of the sodium alginate or not. If not, then I suggest to try this within your new projects so that the efficacy of your 2DR technology would be highlighted.
Very interesting question. So what we know so far is that in addition to the angiogenesis, this is supporting healthy endothelial cells as well. That's basically how the endothelial cells will come and form the new blood vessel formation, and basically helping through this mechanism. There are various other things which we still need to produce more PhDs on this, basically. So we were the first time we have took this 2-deoxy-D-ribose and start investigating and publish so far this data. So yes, so regarding your triple and double layer and triple layer, that's very interesting suggestion. We will definitely think about this.
Doctor, my question is that can we identify the gene that is responsible for the production of this component? And my antithesis would be that if we can regulate that gene, we can also prevent the angiogenesis in diabetic retinopathic patients who have got the proliferation of so many blood vessels on the retina. And another second application would be that instead of using embolic therapy to prevent the vascularization of the tumor, if we can switch off this gene, we shall be able to prevent the angiogenesis in the tumor and reduce its size. Thank you very much.
Very interesting question. I think this project has been originated from the same point of view that on the one side this sugar was triggering the something which we really don't want, but on the other hand side, so this should increase the new blood vessels formations in the critical cases. We need the chronic when this area is big. So yes, for genetic studies, we are still looking to get some funding. So this is on plan. So I'm discussing with the right people who can test this on the gene level. At IRCBM, we have the people which can really help me to study what genes is involved and then more understanding. Yes, that's on plan.
And the factors that switch on and the switch off these genes.
Yeah.
So if we can study those factors, then we can regulate this gene that would synthesize this.
It's a very good suggestion. Thank you, sir.
Okay, this is last question.
Thank you very much, Doctor, sir, for very nice presentation and nice work. I have a question that what is the effect of this sugar on the collagen production in the wounds and also on the adnexal structures regeneration? Does it support the adnexal regeneration or it only promotes angiogenesis? Thank you.
Well, I've shown you a lot of histological study slides when we have tested this on the wound models. First, we have tested this on the simple wound model like 3-4 years ago, and then we were very specific to this more complicated diabetic. That is a well accepted animal model if you're developing something for the diabetic ulcers, basically. So we took the histology, and that's very detailed papers. I will be happy to share with you, or if you can go and see from our website. It's detailed investigations, a lot of HND and CD34 and macrophage 1 and macrophage 2. All the studies. Yes, that's the helping there as well.
Okay, thank you, Dr. Yar, for again very good presentation, and thank you so much. So today, this is the last lecture of today sessions. In this sessions, we covered from own healing to point of care devices and drug delivery systems. So all the presentation were up to the mark, and on behalf of the organizers and the audience, I would like to thank all the speakers of today. They shared their wisdom with us, and thank you very much for listening and taking interest in all these lectures. Thank you so much.